Okay, hello and good afternoon, everyone. So sorry for that little delay. All right. So I hope that you had your wonderful lunch for today. Yeah. So just to check, on everyone, because I don't have my monitor with me. So, ayan, can you hear me loud and clear? Ayan. So can I see a thumbs up button? And if you can hear me loud and clear, tingnan natin kung may magta-thumbs up. All right. Okay, so thank you to those who already put their thumbs up. Okay, so hold on a moment. Yeah. Okay, so before I begin, so I would like to ask first, kumusta yung exam? Yeah, so you can type in the chat box, or you can personally DM me on a range of 1 to 10. So what can you say about the exam? And so 1 being easy and 10 being difficult. Yan. So tingnan natin, no? Just a little survey of, uy, grabe, may 15. May 9.9 na. Ano to, para tayong safeguard. Ayan. Um, may 10. Bakit? Oh, bakit may 50? May 100, 1 to 10 lang. Okay, so that was the last hurdle, no, for you. Ayan, at least for the topic that we already finished. Okay. Um, siguro, no, at the moment, no, yan, kasi nga, you have just finished the examination, no, and some of you may have been, no, still, parang unsure, no, of the answers, and so you still quite don't know kung tama yung sagot niyo o hindi. No? So probably you can reevaluate yourself later on, okay? When we already discussed the answers. All right? Ayan. So, medyo igilid natin to. Para hindi na istorbo yung pagbasa. I will just adjust the screen. Ayan. Siguro ganito. Para nasa isang side yung chat. Tapos nasa isang side yung para makita ko kung meron kayong question. Alright. Hold on. Alright. So, we can now begin. Okay. Um, Ayan. So, this is a 100 item examination covering only topics, no, in biochemistry that we completed last week. No? Ayan. Tapos, actually, no, hindi ko na-mention sa inyo last time that the lectures that I gave you are actually based on the topic outline, no, or based on the objectives, no, that were given by the blueprint of the Professional Regulatory Board, Board of Pharmacy. No? And even the questions that I gave you in the exam, no? So they were based on the number of items, no? Yung item analysis that we say na nakabase din dun sa blueprint. No? So if you did fairly well, no, in this examination, then probably you are quite ready, no, um, to take the exam, right? Ayan. So now let me use the drawing. Okay. And so let us begin, no, answering the questions. If you have any questions, no, and you would like to immediately ask the question, kindly raise your hand, no, or you can type your question in the chat box, right? Yes, I will be constantly monitoring the chat. Yan. Tapos if kunwari din na wala rin yung sound ko for some reason because all of my devices are now charging. And so just do tell me, no? All right. So let's begin with number 1. All cells have the following except. Ayan. So remember, no, that in our review, we talked about the two super domains. No? So we only have the prokaryotic organisms and we have the eukaryotic organisms. No? So in other words, we have the prokaryotic cells and the eukaryotic cells. No? And in our discussion, we mentioned that there are certain parts of the cell that are common, no, in both the prokaryotic cell and the eukaryotic cell. Of course, what separates the eukaryotic cells, no, from your prokaryotic cells would be their presence, no, of membrane-bound organelles, no. Yeah, so normally eukaryotic cells, and normally eukaryotic cells have, let's write it out here. So eukaryotic cells have double membrane. Yan. So meron silang double membrane organelle. Yan. So this is a very general, no, characteristic of your eukaryotic cell. Okay? Yan. All right? So we can now look into the choices that we have. No? So let me just clear the drawing. Yan. Try kong gumamit dito. All right. So all cells have the following. So letter A, cytosol. Of course, yes. No, this is your cytoplasm. So this is the cytoplasm which is present in all cells. All right? Parang naliliitan ako sa size. Tingnan natin. And of course, all cells have the cytosol or the cytoplasm. So that is not our answer. Yeah, next, letter B, genome. Now remember that the genome basically, you know, comprises all the ribonucleic acids or the RNA and the DNA of an organism. So what these molecules are, you know, they are the control centers of your cell. All right. Ayan. So all organisms have their RNA or DNA. Yeah, sometimes both. Okay. So all cells have both. So this is again not our correct answer. Now let's move on to letter B, plasma membrane. Yeah, so the plasma membrane, no, is the membrane, no, that keeps the cell intact. No? And remember that the plasma membrane or the cell membrane, yan, pareho lang yun, the plasma membrane or the cell membrane is different from the cell wall. Okay? Yan. So which organisms have the cell wall? Yeah, so remember that only your prokaryotic cells, your prokaryotes in general, both your eubacteria and your archaebacteria have the cell wall. Okay? Yeah, so medyo mag-review lang tayo ng konti, no? While we rationalize this question. Yeah, so remember that the archaebacteria, no? Yeah, so archaebacteria or the family, the domain archaea, no? They have pseudo peptidoglycan membrane, okay? And yung pseudo peptidoglycan membrane nila would consist of, ayan, do not ever forget this, no? N-acetyl tallose aminoronic acid. Aminoronic, yan, hindi acid. Okay? And N-acetyl glucosamine. Okay? Ayan. So, HDLC. Yan. So, N-acetylglucosamine. Alright? Now, for the bacteria, eubacteria, so yung eubacteria naman, it has the murine. Okay? The murine, which basically consists of peptidoglycan, and the peptidoglycan consists of N-muranic acid. So, let's figure out where to write. N-muranic acid, yan, and N-acetyl glucosamine. Right? Yan. So, what differentiates your archaea from your eubacteria ay yung presence of N-acetyl tallose aminoronic acid in place of N-acetyl muramic acid. Okay? In your eubacteria. Okay? But then again, those are components of the cell wall. Aside from your prokaryote, sino pang organism, no, in the eukaryota, no, in the superdomain eukaryota, yung mayroong cell wall, it's your fungi. Okay? Ayan. So remember that fungi are eukaryotic organisms. No? They have eukaryotic cells and they have cell wall. Okay? Tapos yung kanilang cell wall, no, ay composed of chitin. Okay, yan. So, ang laman niyan ay chitin, mostly chitin. And then, of course, we have plants. Okay? So, yung plants also have cell wall, and that's the reason why plants are able to survive, no, in high humidity or high water content environment. No? Ayan. So, it's because of the presence of the cell walls in plants. Okay? Ayan. So, yeah, that's basically no those three choices. So that means let me erase everything. Okay. Yeah. So that means our answer will be letter C, mitochondria, right? And so letter C, mitochondria. Yeah. So remember that the mitochondrion, right? The mitochondrion is a membrane-bound organelle, which only exists in your eukaryotic cells, right? Ayan, liitan natin na lang ito. Okay. Ayan, all right? Now, regarding the mitochondrion, okay, so remember that the mitochondrion is the organelle where oxidative phosphorylation occurs, no, in eukaryotic organism. However, pagdating natin sa mga prokaryotic cells, okay, the oxidative phosphorylation occurs in the plasma membrane. Okay? Yan. So that's why your prokaryotic cells do not require or do not need the mitochondria, no, for the oxidative phosphorylation part. All right? Yan. So number 2. Okay, so now let's proceed. The macromolecular complex that can be found in the organelle where protein synthesis occurs is the, ayan. So for this one, no, we are asked to determine where protein synthesis occurs, no, and of course, our answer here is letter B, the ribosome. Okay, and so C, the ribosome. Okay. Now, your lyoxysome and your spirosomes, these are membrane-bound organelles, okay, that are responsible, no, for the synthesis of carbohydrates and lipids, okay, in certain organisms, some in plants and sometimes in both plants and animals, okay. And then the lysosome, no, is the organelle wherein there is degradation, no. This is called the waste basket, no? And this is called the waste basket of your cell, no? Ayan, lysosome. So it's called the waste basket of the cell because um, it is where the um, the tawag dito, biomolecules which are supposed to be disposed, no, are placed. Okay, yan. And sometimes the lysosome is also responsible for the degradation of invading pathogens like your viruses and bacteria. All right, yan. So that's for number 2. All right, so next we have number 3. Protein processing, this is the key term here, protein processing, such as modification and synthesis of the carbohydrate portions of the glycoprotein occurs in this complex of vesicles and folded membranes within the cytoplasm, yan, of most eukaryotic cells. Yan. So now remember that protein processing only occurs in eukaryotic cells. Okay? Kasi nga, one other difference between your prokaryotic cell and your eukaryotic cell is that yung eukaryotic cell ay capable, no, of separate replication, transcription, and translation. And not only that, no, once the protein has been translated, nagkakaroon tayo ng protein processing, no? So, for example, addition of sugar moieties, no, sugar groups, no, in the proteins to produce the glycoproteins, and so that only occurs in your eukaryotic cells. In prokaryotic cells, now remember that replication, transcription, and translation can occur simultaneously. No? And because of that, there can be no modifications to the mRNA transcript or even the protein. Okay? Ayan. So, kaya yun yung important no, in this item. No? So, yun yung i-take note ninyo. So, let's look at the answer. So, letter A, endoplasmic reticulum. And so, remember that in the endoplasmic reticulum has two types. Okay? So, you have the smooth endoplasmic reticulum and you have the rough endoplasmic reticulum. Okay? So remember that the smooth endoplasmic reticulum is where fats are produced. Okay, yan, fat. Kasi nga smooth, di ba? Parang smooth skin, no? Very oily. Yan. And then rough endoplasmic reticulum is where proteins are produced. Okay? Ayan. So next is the letter C. Punta muna tayo kay letter C. Lysosome, ayan, nasabi na natin kanina, this is the waste basket of your cell. Then letter D is the mitochondrion. This is the powerhouse of the cell, no, where energy no, is produced. Most of the energy is produced through oxidative phosphorylation. Okay? Yeah, so our answer here is letter B, no, the Golgi or the Golgi complex. All right? And so letter B. Okay, so just tell me, no? You can raise your hand or you can type your question in the chat box if you have any question. Ayan. All right, so now let's proceed to the next number. Ayan. So number 4. Blank make up centrioles in a cell, and they are the base of both the flagella and cilia, okay, of the cell. Yeah, so letter A is intermediate filament. Letter B is microfilament. Letter C is microtubule. And letter D, myosin fibers. Okay? Yeah, so we have heard of the myosin fibers, no? Yeah, probably in your human physiology and pathophysiology class, no? The myosin fiber is the fiber that is present in your contractile muscle fibers. Okay? So sa muscles ito nakikita. Ayan. And I mentioned the myosin, no, when we discussed the myoglobin. Okay? Ayan. So, medyo related sila. They are both no, in the skeletal muscles. Now, we are only left with the following. No? So, we have the intermediate filaments, the microfilaments, and the microtubules. Ayan. Now, remember no, that the microfilament is also known as the actin filament. No? The actin filament no, is the smallest filament among the cytoskeletal elements. No? Ayan. So, the three, A, B, and C, no, the intermediate filaments, microfilaments, and microtubules are cytoskeletal elements. No? From the largest, no, remember that we have the microtubules. Ayan. Ito yung pinakamalaki. Ayan. Followed by the intermediate fibers and then the micro filaments, no, yung smallest of them. Okay. Now, movements no, in the cell no, can occur because of your microfilaments. Okay? Yan. So, yung cytoplasmic streaming. Okay? Yan. So, that occurs because of the presence of the microfilament. Your intermediate filaments can also assist no, in the structural integrity of the cell. Okay? But it is the microtubules no, which is responsible for the scaffold and for locomotion. Ayan. So scaffolding and locomotion is for the microtubules. And last time, no, I mentioned na the flagella or the flagellum, no, pag kung singular lang, consists of tubulin molecules, no, or tubulin fibers, no. And it is the same molecule that is present in your letter C, microtubules. So that is our correct answer. All right. Letter C, microtubules. All right. Number 5. Yeah, so for number 5, the peptidoglycan in the eubacterial cell wall contains alternating units of N-acetylglucosamine and blank, which are cross-linked by oligopeptides. Ayan. So I already mentioned this before, no? So our answer is letter B, N-acetylmuramic acid. Okay? And in fact, no, because of N-acetylmuramic acid, present in your peptidoglycan is also known as murine. Murine. Okay? Ayan. So that's your peptidoglycan. Okay? Next is number 6, which is a membrane-bound cell organelle that contains digestive enzymes involved in the degradation of various biological polymers, excess or worn out parts, and even foreign entities like bacteria or viruses. Ayan. So, for this one, no, binalikan ko kasi sabi ko, parang naulit tong tanong na to. Hindi pala. Ayan, so, this is my explanation no, in the previous number. Okay, so yung number, yeah, sa number, number Ayan, 2, di ba? Ayan. So sa number 2, in-explain ko dyan that the lysosome is also known as the waste basket. No? Ayan. So it is the waste basket of the cell. Okay? Ayan. So for number 6, no, our answer is letter A. Ayan. Lysosome. Lysosome. Yeah, so again, no, balikan natin yung ibang mga choices. Um, na, now that we know that lysosome is the waste basket, no, where materials that need to be degraded will be thrown into, ayan, the other organelles here are letter C, peroxisome. No? So si peroxisome, this is an organelle which contains peroxidases. Okay? Ayan, so pag sinabi nating peroxidase, ayan, peroxidase, ayan, this is an enzyme that hydrolyzes your organic peroxides. No? Ayan. So, nai-inactivate niya, no? Your peroxidase inactivates organic organic peroxides. All right? Now, a special type of peroxidase is actually your catalase. So, peroxidase and catalase, no? So, catalase naman is an enzyme, no, which catalyzes the hydrolysis of your hydrogen peroxide. Okay? Yan, yung catalase ay para sa H2O2. Okay? Yan, yung peroxidase naman ay para sa ROOH. No? Ayan. So, peroxides. Okay? Ayan. Now, yung catalase, no, this inactivates hydrogen peroxide to form water and oxygen. So, water and oxygen. Now, take note na both peroxidases and catalases, no, are important enzymes for the survival of aerobic organisms. Ayan. They are needed by your aerobic organism. Okay? And they can be found in your peroxisome. No? Ayan. So that is not our answer. Letter D, ribosome. This is not an organelle. Ayan. So i-highlight natin yon. The ribosome is not an organelle. This is only a macromolecular complex, no, containing proteins and RNA. Okay. Now the ribosome is where protein synthesis occurs. All right. Ayan. Parang ang daming umaalis. Ayan. Okay. So letter B, nucleosome. Nucleosome is it is the complex. No, it is also a complex. It's a complex of the DNA and histone. Okay. Ayan. So, DNA, yan, DNA plus histone molecules. And there are, remember, 8 histone molecules where the DNA winds itself into, no? And that is what you call the nucleosome, no? So, for number 6, our answer is letter A, lysosome. All right? Number 7, ayan, which is a unique characteristic of eukaryotic cells. Ayan. All right, so let's begin with letter D. No, yeah. So letter D, protein synthesis in the ribosome. No, is this a unique characteristic of eukaryotic cells? The answer is, yan, may gusto ba mag-participate? Yan, para hindi sila umaalis. Ano yung malis eh? Oh, yun, no? Mga taga Yupang. Okay. Ayan. So, yeah. Okay, so probably they just have a very difficult time logging. Okay, so the protein synthesis, no, of course, occurs in all organisms, no, and they all occur in the ribosome. Okay, yeah. So this is not our correct answer. Letter C. metabolic reactions occurring in the cytosol. Okay. Now, remember that the prokaryotic cells technically don't have the nucleus. No? Yeah, so the prokaryotic cells don't have the nucleus. The prokaryotic cells don't have the mitochondria. Okay? So, ibig sabihin, wala na siyang choice. Yan. So, the prokaryotic cells can only have their metabolic reactions in the cytosol or in the plasma membrane. Okay? So, this is again not a unique characteristic of your eukaryotic cells. All right? So letter B, and coupled transcription and translation. Yeah, so remember that eukaryotes have compartments, okay? And because of the compartmentalization of the cell, okay, different reactions occur in different compartments or in different organelles. So there can be no coupled transcription and translation in eukaryotic cells. Okay, in fact, for eukaryotic cells, no, yung mRNA transcript, okay, so remember that the mRNA transcript na lumalabas in the DNA, no, is actually called pre-mRNA, no? And this pre-mRNA, no, then goes out of the nucleus, no, and it is being spliced, no, by the spliceosome, which cuts off the introns from the exons, okay, to produce the mature mRNA. And that is an example of a modification, no, a post-transcriptional modification, which only occurs in your eukaryotic cells. Okay, and there can only be post-transcriptional modifications if the transcription and the translation machineries are uncoupled. Okay? So letter B is not our correct answer. So our correct answer is letter A. Ayan. Cellular communication via gap junctions. No? Ayan. So remember that bacteria, no, even though they don't have gap junctions na tinatawag, okay, for cellular communications, bacteria also have a certain type of cellular communication. Okay? Ayan. Pero bakit ba muna walang cellular communication yung bacteria through the gap junctions? No? Because technically prokaryotes are unicellular organisms. No? Yan. So what I am the style that I am doing with you sa rationale ay pinapakita ko sa inyo that even if you don't know the answer right off the bat, no, agad-agad, no, kahit na hindi mo alam yung sagot agad-agad, no, you can actually conjecture, no, or you can have a scientific guess, no, um about the correct answer. Okay? Yan. And the only way to do that is to eliminate the choices. Okay? Yan. So, sana naka-follow nyo yung explanation of why the other choices are wrong. Okay? And although there are, of course, certain items na madaling makita talaga yung tamang sagot, okay? There are choices where you really need determine why others are wrong. Okay? Ayan. But for letter A, you have to think of this, like this, Remember that prokaryotes are unicellular, All prokaryotes, without exception, all of them are unicellular, And unicellular organisms do not have gap junctions, okay? Kasi yung gap junctions, ito yung present, in your eukaryotic cells where there can be cellular communication, okay? Yan. So yan yun, in between the two cells. Ngayon, yung mga bacterial cells mo, hiwa-hiwalay siya. No? Ganyan. Yan. So, hindi siya magkadikit. Okay? Yan. So, dahil dyan, wala siya talagang gap junctions. Alright? But, yan. So, bigyan natin ng malaking but. And remember na, for prokaryotic cells, they can have intercellular communication through a process that we know as quorum sensing. Ay, ito yung magandang pen. Quorum sensing. Ayan. So, ito yung mukhang galaxy. Ayan. So, quorum sensing. Alright? And this one, is responsible for the formation of what you call the biofilm. Okay. Ang ganda nito. Ay, isa pen. Okay, so this is the biofilm. Yan, biofilm. Okay, yan. So, once prokaryotes no, communicate no, to come together, they aggregate to form a biofilm. Okay? Ayan. So, the answer for number 7 is letter A. Okay, try natin tong pen na ito. Okay, number 8. So as you can see, there are 7 items no, related to the cell. No, introduction to biochemistry and the cell. Okay, next item. Ayan, so number 8. Which of the following statements about fatty acids is true? Ayan. So for this type of questions, no, the best thing that you can do is to eliminate the choices. Okay? Kasi minsan, no, hindi madaling makita kung ano yung tama at saka yung mali. Alright? So let us begin with letter D. No, ayan. So letter D, only saturated fatty acids can be found in the triglycerides of mammals. Okay? At color pala siya. Okay lang ba to? Nakikita niyo naman. Sige, palitan natin. So, tatabaan kasi ako dun sa kanina. Okay. Ito na lang. Only saturated fatty acids can be found in the triglycerides of mammals. Okay? Now, the thing is, again, meron tong thing. Yeah, so the thing is, ayan, dapat pala hindi pa ako dito nagsimula. Okay. Okay, so anywho, okay, tama. Okay, sige. Letter D. Panindigan natin yung letter D. So, letter D. Only saturated fatty acids can be found in the triglycerides of mammals. This is my hint for you, no, for the exam, no. Whichever kind of exam that is that, no, that you are going to take, if you see, no, if you see the terms all, only, yung mga generalization terms, okay? Like all or only, no? Ayan. So yung mga yon, okay? Those are actually markers na mali yung sagot. Okay? Ayan. So those are markers na mali yung sagot, okay? Kasi once you generalize that everything has to be one thing, no? Sometimes there are exceptions. Okay? Ayan. Kaya kadalasan they are wrong. Okay? So for letter D, this is actually wrong. Okay? And so remember that we know that saturated fatty acids, yes, no, they can be found in fat. Okay? Yeah, so remember that saturated fatty acids can be found in fats. No? And we know that fats come from animals, no, particularly mammals. Okay? Now, even though fats, no, which contain saturated fatty acids can be found in mammals, okay, that does not mean na only no, saturated fatty acids can be found in mammals. No, in fact, no, there are other no fatty acids, no, unsaturated fatty acids that can be found no in animal na in animal fat, no? Yan. So for example, no, yung mga beef tallow, no, and yung pork fats, no, they would often have other, no, unsaturated fatty acids. So because of the term only, no, this item is wrong. Okay? Also remember, no, na yung mga fish oils naman, although they are not mammals, no, yung fish oils, they contain mostly unsaturated fatty acids, no? Ayan. And then kung babalik rin naman natin, no, remember na even plants, no, there are plant sources of your saturated fatty acids, no? Ayan. And because yung mga plants na ito have a lot of saturated fatty acids, no, they tend to be solid at room temperature, no, just like the fats of mammals, right? So letter D is wrong. Talon tayo, no? Ayan, talon tayo sa letter B. Okay? Ayan, ito yung sinasabi ko kung bakit nagkamali ako ng sinimulan. So letter B, ayan, in naturally occurring fatty acids, the groups around the double bonds are always in trans configuration. What makes this statement wrong is is the word trans. No? Ayan, so remember na trans trans fats, no? Trans fatty acids are not easily metabolized by the body, no? Ayan. So they are not, no, supposed to be present in naturally occurring fats and oils, no? Ayan. What is common, no, in naturally ay yung cis configuration. Okay? The trans configuration of fatty acids, no, can only happen, no, kapag semi-synthetically produced yung fats and oils. Okay? But not in naturally occurring fatty acids. All right? So letter B is also wrong. So punta tayo kay A. Yung A mukhang tama, no? Ayan. Both oleic and linoleic acids are considered as essential fatty acids. Now remember, when we say essential fatty acids, these are fatty acids, no, which are to be taken from the diet. No? They cannot be produced by the body. Unfortunately, C18-1, which is oleic acid, is produced by the body. Okay? Ayan. So, for that reason, no, this statement is wrong. Okay? So, sino-sino ba yung mga essential fatty acids? No? So, remember, you have your linoleic acid, linolenic acid, you have your DHA, EPA, and DPA. No? So you have your docosahexaenoic pentaenoic acid, docosahexaenoic acid, and eicosapentaenoic acid. Those are all essential fatty acids. No? And the most common, no, the most common essential fatty acid source would be your fish oil. Okay? Yeah. So letter A is also wrong. So that means the answer is letter C. Ayan. In the endoplasmic reticulum, double bonds are introduced between C9 and C10 of saturated fatty acids by the fatty acid oxidase. No? And remember, I mentioned this to you, no? Na, yan pa pala. Ayan. So remember that I mentioned this to you na si delta 9 desaturase, no, is another enzyme. No, it's the same enzyme actually. That introduces the double bonds, no, in your saturated fatty acid. All right. Yan. So are we clear? For number, ang dami umalis. Oh. Parang gusto kong i-monitor kung ilan yung mga tao ngayon. At saka, ayun, no, 60 na lang. Dito na lang tayo. Baka perfect nila yung exam. Okay. Oh. Medyo nababother ako pag may umaalis.
Kaya,
Nakikita ko dito.
nung tignan yung lecturer na mismo, yung problema.
Sabi ko, parang kanina medyo marami po sila. Okay, so anyways, ayan, let me proceed to number 9. Okay, fats and oils from animals and plants are considered as, okay, ayan, so let's look at the choices, no? Ayan, so letter A, ayan, bile acids, letter B, gangliosides, letter C, phosphoglycerides, or letter D, triacylglycerol. Ayan, all right. Now, remember, no, yung what I drew, no, on the board last week, no, that we can actually classify your lipids, no, yan, into several classes. Okay? Yan, kasi nga remember that lipids, no, yung mga lipids, they are not really they are not really polymers. No? They are not biopolymers. Okay? Yeah, because they are not biopolymers, no? They don't have the same, okay, structure. No? Ayan, wala silang common na structure talaga. Okay? Ayan, in fact, what makes your lipids, no, lipids would be two properties. No? So you have the hydropathy, no? So ibig sabihin they do not mix well with water, okay, or they are water insoluble. And their lipophilicity, no? They're being soluble in fats, no? Yan. So lipophilicity, okay? Yan, or fat solvents, no? So our fat solvents would be your hexane, your diethyl ether, petroleum ether, no? Even alcohol and acetone, no? Yan. So those are your lipids. Okay? Now, yung drawing natin last time was like this. Okay? So remember, pag sinabi nating lipids, ayan, so we have here the fatty acids, no? The fatty acids plus the glycerol, no? So ito yung ating tinatawag na triglyceride. Ayan, so meron tayong triglycerides. And remember that triglycerides are composed of free fatty acids, ayan, free fatty acids, and meron din siyang glycerol. Okay? Ayan. Pero pag sinabi kasi nating triglycerides, there should be three fatty acids present, no, in the molecule. Okay? Ayan. So triglycerides are also known as acyl glycerides. Ayan. So remember na yung ating triacyl glycerides, no, can be classified into two, no? So pwede siyang simple triglycerides, no? And we have the mixed triglycerides. Ayan. So pag sinabi nating simple triglycerides, these are triglycerides containing the same fatty acid in all the three positions of your glycerol, no? Yan, kasi this is our glycerol moiety. Yan, so CH2O, tapos ito yung fatty acid component, no? So R, CH, COR, and then CH2O, COR. Okay, ayan. So if all the R groups are the same, then you have a simple triglyceride. Tapos mixed triglyceride kapag iba-iba, no? Ayan. For example, iba ito, iba yun nandito. Okay? Yan. So you have a mixed triglyceride. And now, when we discuss the fatty acids, remember that we discussed two types of fatty acids. You have saturated fatty acids. So, yeah, saturated fatty acids. And you have the unsaturated fatty acids. Ayan. Okay? Now, next one, no, na type ng lipid will be the phospholipid. Ayan. So, we have the phospholipids. Ayan. So, yung mga phospholipids natin ay meron tayong phosphatidyl, phosphatides. Okay. We have the phosphatides. Yan. So pag sinabi nating phosphatides, ito sila phosphatidyl ethanolamine, no? Phosphatidyl ethanolamine, tidyl, yan. Lagyan natin ng ethanolamine, which is acetylene. Yan, cephalene. As this is your phosphatidyl choline, this is your lecithin, choline. This is your lecithin. And meron din tayong phosphatidylserine, tidylserine, and that phosphatidyl inositol, and in fact meron pang isa, no? Ayan, phosphatidyl glycerol. Yan. So remember na for this one, okay, one um one group, no? So ito will be replaced by phosphodiester bond, no, which is bonded, no, to an alcohol, no, and the alcohols can be any of the following: ethanolamine, choline, serine, inositol, and glycerol. Yan, bago ko makalimutan, ilagay na lang din muna natin na phosphatidyl inositol is also your inositide. Ayan. Okay? Yeah, so these are your phosphatides. Okay? Now, remember na if we remove um, a fatty acid from either position 1 or position 2 of your glycerol in a phosphatide, no, it now becomes a lyso. No? Lysophosphatide. Yan. Alright? Yan. So, nagiging na siyang lysophosphatide. Okay? Ayan. Now, if yung pangalan niya will depend on the alcohol, no, that is present in the phosphodiester bond. So for example, you have lysophosphatidyl inositol, lysophosphatidyl choline, and so on and so forth. All right. Now for the aside from phosphatides, no, meron din tayong other types of phospholipids. Okay? Yan. So, for example, we have your um, cardiolipin and your plasmalogen. Plasmalogen. Alright? So remember na pag sinabi natin cardiolipin, it is diphosphatidylglycerol, no? Glycerol. Okay? So ibig sabihin, there are two phosphatidic acid residues, no? So two phosphatidic acid residues and one glycerol. Okay? So nasa gitna yung glycerol and then you have the two phosphatidic acid residues, no? So it's like your phosphatidylglycerol na dinagdagan mo pa ng isa pang phosphatidic acid. All right? And that's your cardiolipin. Remember that your cardiolipin is the antigenic, no? It's the antigenic na phosphatide, okay, or phospholipid. And then you have your plasmalogen. Well, plasmalogen is very much similar to the phosphatide, except that you don't have the ester bond, no? Ayan. So you don't have the ester bond. Ay, ibay natin yung full. Undo. Okay. You don't have the ester linkage, no, in S1 or C1 position of your glycerol, no? Instead, you have an ether link, no, at that position. All right, that's your plasmalogen. Tapos, in the diagram, no, we also have, ayan, aside from phospholipids, you have your glycolipids. Ayan. So, yung ating mga glycolipids naman, ito sila cerebroside, meron tayong sulfatide, at saka si ganglioside. Ayan. Pag sinabi nating cerebroside, no, this is a sphingolipid, no? Ayan. So, meron tayong sphingolipids, which I forgot to mention here. So, sphingolipids muna. Sphingolipids. Ayan. So, pag sphingolipids, meron tong sphingo, no? Sphingosine. Ayan. Sphingosine. And then, if the sphingosine has a sugar unit, ayan, doon na tayo nagkakaroon ng mga glycosphingolipids. Okay? Yan. So you have your cerebroside which contains monohexoses. Yan, you have your ganglioside which has the sialic acid, no? Particularly neuraminic acid. All right? Yan. So yan yung mga lipids. Now, meron pa actually ako nakalimutan, no? Although hindi ko nakalimutan, like ako lang idagdag. You also have your vitamins, no? Your fat-soluble vitamins, no? So vitamin. And these are your ADEC, no? Yan. So ADEC and steroid. Ayan. Okay, so sobrang varied, no, na structure of your lipids. All right? Kaya nga, no, kailangan nating maisip, no, yung classification of your lipids. All right? So let us now go back to the question at hand. Ayan. So number 9, fats and oils from animals and plants are considered as letter D. Okay, so they are triacyl glycerol. Okay? Yan. Remember that bile acids, these are actually steroids in structure. Ba't ang kapal ulit? These are steroidal in structure. And your gangliosides, no, are glycosphingolipids. Your phosphoglycerides, no, these are your phosphatides. All right? Number 10, which is a saturated fatty acid. Ayan. So remember na that I mentioned to you how to name, no, your fatty acids. Okay? If you have a simple, naming of triglycerides and fatty acids, no? So punta muna tayo sa triglycerides, no? For triglycerides, no, if you have a simple triglyceride, okay, and it contains only the same, no, fatty acid in all the three alcohol groups of glycerol, no, ang pangalan niya will simply be the name of the acid, no? Ayan, let's say palmitic acid, okay? But we drop the ic acid, okay? So for example, no, yung tatlong fatty acid ng inyong triacyl glyceride, no, or ng inyong fat or oil ay palmitic acid, no? We drop the ic acid and then we replace it with in. No? So palmitin. Okay? Yan. So anong ibig sabihin nito? Your fatty acid here is your palmitic acid. Your triglyceride is palmitin. Okay? So in other words, yung palmitin dyan is already wrong. Okay? Next. So na-eliminate na natin letter D. Okay? Kapag mixed triglyceride naman siya, no, you have to name all the fatty acids. Okay? Ayan. So you have to name all the fatty acids, and the longest fatty acid will take the final or the base name. All right? Okay. So next would be these other choices. No? Ayan. Tingnan natin si letter B. Letter B appears to have IN din in the suffix. Okay? So that means palmitolein is also a triglyceride. Okay? Yeah, so sino naman kaya yung fatty acid ng palmitolein? Okay? So palmitolein ang kanyang fatty acid is palmitoleic acid. So palmitoleic acid. All right? Ayan. So remember yung ating code, no? Yung code natin before, yung CLM, okay? PSA. Yan. So this is your C10. This is your uy, mabagal tayo. This is your C10. This is your C12. Yan, this is your C14. This is your, this is C. Yan, C16. Good. Tapos this is your C18. And this is your C20. Okay? Ayan. So this is your capric acid, okay? Capric acid, lauric acid, myristic acid, palmitic acid, stearic acid, and arachidic acid. All right? Now, palmitic acid has 16 carbon. Now, if it has one unsaturation, it becomes palmitoleic acid. Okay? Ayan. Kasi remember, oleic acid is C18 with one unsaturation at carbon 9. Okay? Yan. So, ganun din si palmitoleic acid. No? So, it has one unsaturation at carbon 9. Okay? But, it is only a C16 fatty acid. Right? So, that's your palmitoleic acid, which becomes your palmitolein. Yan, palmitolein. Okay? Ayan. So, that being said, no, letter B is also wrong. Yeah, so, dalawa na lang yung choices natin. Letter A and letter C. Okay? So, na-mention ko na kanina, your palmitic acid, no, is a saturated fatty acid kasi nandito nga siya. Okay? It's a C16 saturated fatty acid, while your palmitoleic acid is a C16 unsaturated fatty acid. So our answer for number 10 is letter C, palmitic acid. Okay? Are we good? Okay. Ayan. So next we have number 11. And which of the following fatty acids is not an omega-3 fatty acid? Ayan. So for the omega-3 fatty acid, remember that these are essential fatty acids, which are commonly derived from fish oil. Okay. Now, as mentioned before, okay, so the DHA and EPA are essential fatty acids, and they are in fact also omega-3 fatty acids. So A and B, A and B are not omega-3 fatty acid. They are omega-3 fatty acid. Okay? So, gawan ko ng check. Yan. So, they are omega-3 fatty acid. Ang tanong, not. Okay? Yan. So, naiwan sa atin si alpha linolenic acid at saka si gamma linolenic acid. Okay? So, remember that for these two, so, di ba may tatlong magkakasama yan. Si oleic, linoleic, at saka si linolenic. Yan, ba't ang bagal ng... Okay. So, you have your oleic, linoleic, and linolenic acids. Alright? So remember that oleic acid is 18-1. Okay? Ayan. So 18-1. So ibig sabihin 18 carbons, 1 unsaturation at carbon number 9. And it is this. Okay? Si linoleic acid naman, no? Yan. So as mentioned before, linoleic acid. So may oleic siya. So siya yung pinakamalapit kay oleic. Okay? Ayan. So this is your 18-2. Okay? 18-2. Ayan. And then the two unsaturated bonds are present at carbon 9 and 12. Ayan. Okay? Next, we have the linolenic. Okay? So now remember that there are two linolenic acids, alpha and gamma. Linolenic acid. Ayan. So for the alpha linolenic acid, it's 18-3. Ayan. Tapos yung gamma, it's also 18-3. Okay? There are three unsaturated bonds. Okay? And they have 18 carbons. Okay? Now, for alpha-linolenic acid, remember na, gagayahin lang natin yung mga nauna sa kanya, no? 9C, 12C, and 15C. Okay, ayan. So, yan yung mga double bonds. No? They are present all in cis configurations at carbons 9, 12, and 15. Okay? Now, yung gamma, linolenic acid, no? The positions of the double bond will be at 6, 9, and 12. Okay? That being said, we have identified na si, yan, palit tayo ng color, na si alpha, no? Alpha-linolenic acid is an example of an omega-3 fatty acid. Bakit? Kasi there are 18 carbons in the molecule. The last double bond is present at carbon 15. Okay, yan. So, 18 minus 15 is 3. No? Kaya siya omega-3 fatty acid. Now, for the for the 16 na lang tayo. For the gamma linolenic acid, okay, the last double bond, the last unsaturation is carbon 12. Okay, there are 18 carbons in the molecule. So this is an example of an omega-6 fatty acid. All right? So yung ibig sabihin ng omega-3, omega-6 would be the position of the last unsaturated bond. Okay? From the terminus. Okay? From the alkyl terminus. So our answer for this, no, which is not an omega-3 fatty acid, is letter D. Ayan. Gamma linolenic acid. Because C, letter C, is an example of an omega-3 fatty acid. All right? Pencil pala yun. Palitan na. Okay. Ito na. Hold on. Nagkapalit lang ako ng kulay ng pen. All right. Okay, so if you have questions on just don't hesitate to ask. Number 12, which are true about the fatty acid with this abbreviation? 18, 3, 6, 12, and 15. Yan. So of course, this is your gamma linolenic acid. Okay? Ayan, so nakita na-explain ko na kanina no kung sino si gamma linolenic acid and what these numbers actually mean, no? But just as a review again, no, 18 here is the number of carbons in the fatty acid, 3 is the number of unsaturated bonds, okay? The position of the unsaturation will be 6, 12, and 15, and the configuration is cis, right? Ayan, so let's see, no, all the choices. By the way, no, in the board exam, usually they would have like questions like this. Okay, 1, 2, 3, 4, and then you have the choices. Okay, so you have to be able to determine kung true or false sila. Right? So number 1, the fatty acid has 18 carbons. Ayan. So this is correct, no? Ayan, the fatty acid has 18 carbon. Next, the fatty acid is a saturated fatty acid. So this is wrong. Okay? Kasi nga, sabi natin, unsaturated siya. Okay? Number 3, the fatty acid has 3 double bonds at C6, C12, and C15. This is also correct. And number 4, the fatty acid has a triple bond. This is wrong. Okay? In fact, there is no naturally occurring fatty acid with a triple bond. Number 5, the configuration around the double bonds is all cis. Okay, so this is also correct. So our answer is 1, 3, and 5, no? And that is letter D. Ayan. Okay. Number 13. The structure below is an example of a blank. Ayan. So let's look at the structure. This is actually the glycerol moiety. No? So this is an example of a glyceride. Okay, we just don't know kung anong klaseng glyceride. Now, if we look at the other portions of the molecule, no? So for example, this one, this is the phosphate group, and ayan, meron siyang alcohol. Ayan. So ito yung alcohol moiety niya. Okay? Now, this alcohol moiety is actually your, ayan, this is your ethanolamine. Ethanolamine. Okay, yan. So that's your ethanolamine. Yan. Now, so it appears na this is a phosphoglyceride, no? Hindi lang natin alam anong klaseng phosphoglyceride. Now, the the other group, no, that we have to look into ay itong group na ito. Yan. So this is an ester bond, no? Yan. So this is an ester bond, O, C, O. Ayan. So this is the fatty acid. Okay? Yan. So that's a fatty acid. So you have a fatty acid, you have a glycerol moiety, you have ethanolamine, and a phosphate group. Then, finally, no, meron tayong isa pang group na nandito. Yan. Okay? And if you look at the bond, no, present in this group, itong bond na ito, no, this is actually an ether bond. Okay? Yan. So, in other words, this one is a letter C, plasmalogen. Okay? Ayan. So, kasi ayun yung kakaiba sa kanya, no? So, on carbon 1 of glycerol, it has an ether bond rather than an ester bond. Okay? Ayan. So, letter C, plasmalogen. Ayan. Number 14. What do you call a phosphoglyceride containing only one fatty acid in either C1 or C2 of glycerol? Ayan. So, na-mention natin to kanina, no? Kapag, no, merong kang isang phosphoglyceride, no, having the glycerol moiety, no? So, you have here O, tapos H na ito, tapos over here, ay, sige lang, kulang ako ng carbon. Phospho. Ayan. O. Tapos dito yung phosphate. Ayan. So, nandito yung phosphate group P, O, O. Tapos this is an alcohol group R. Tapos you have here the fatty acid, no? Either dyan or pwede rin sa kabila, no? Pwede rin dito. Okay? Ayan. So, this is an example of a letter B, lysophosphatide. Okay? Ayan. All right. Number 15. Yan, lecithin is a complex, naturally occurring mixture of phosphatide. It is also known as, ayan, so sinabi na natin to kanina, no? So if you were just listening, ayan, so tingnan natin. Ayan, so 59 na lang tayo. Okay, lecithin is a complex, naturally occurring phosphatide, and it is your letter A, phosphatidylcholine. Okay? Number 16, which of the lipids below are synthesized in the liver and contain a steroidal backbone? Ayan. So that is letter A, no? Bile acid. No? So yung iba dito na mention ko na, no, when I introduced to you the schematic diagram. Okay? Number 17, blank are derived from 5-hydroperoxy eicosapentaenoic acid or 5-HPETE and modulate a variety of biologic functions. Okay, so remember that your prostaglandins, thromboxines, prostacyclins, no, and leukotrienes come from the same parent. Ayan, from the same parent molecule, no, or precursor, and that precursor is arachidonic acid. Okay, so all of them come from arachidonic acid. Now there are two pathways, right? So you have the COX and you have the LOX. No? So you have the cyclooxygenase pathway and the lipoxygenase pathway. All right. So yung cyclooxygenase pathway, there are two, like cyclooxygenase pathway. You have the COX-1 and you have the COX-2. Okay. So na-mention natin before that COX-1 is ubiquitous, no? It is an enzyme that is capable of catalyzing the production of prostaglandins which are needed, no, by the body. Okay? They are ubiquitous, so ibig sabihin they are present at all times. Okay? One of the most important functions of your ubiquitous prostaglandins would be the production, no, of the mucous membrane in the gastrointestinal tract, no, to protect the stomach, no, from the gastric acids. Okay? Kaya nga inhibition of COX-1, no, lalo na if you have non-selective COX inhibitors, would also cause ulceration of the stomach, no. Now, COX-2, no, leads to the production of prostaglandins, which are inflammation and pain mediators. Okay, and they are only present if there is trauma, if there is inflammation, no, in the tissues. All right. Now, this produces your prostaglandins and your thromboxanes, and your prostacyclin. Okay? Sa cyclooxygenase pathway sila. All right? Now, in the lipoxygenase pathway, sa LOX, no, ang dinadaanan muna nito ay si 5-HPETE. No, ayan. Oopsie. Sana hindi siya bumaba. Ayan. So, your LOX, no, produces your 5-hydroperoxy eicosapentaenoic acid or 5-HPETE, and this is the direct precursor of your leukotriene. No, yan. So, leukotriene. Yan, or LTs pala. Leukotriene. L, yan. So, the answer for number 17 will be letter B, leukotriene. All right? Ayan. Okay. Now, let's proceed to numbers 18 to 20. And an 85-year-old woman complained of excessive stomach ache after taking meals. A couple of months ago, she had been rushed and to the ER due to myocardial infarction. She mentioned that on the way to the hospital, she was given an OTC drug. After some therapy, she recovered and then she was discharged. And fearing that she would have another heart attack and she continued taking the same drug every morning prior to current complaint of stomach. Hold on a second, meron lang ako sasagutin. Yan, okay. So sana naintindihan nyo na yung question, no? With a short break, no? Ayan. So number 18, ayan, the biosynthesis of which lipid may have been disrupted by the continuous intake of the drug. No? So let us analyze what the problem really was. No? There is excessive stomach ache, which led to the chief complaint, no? Why the patient, teka lang, wait, yeah. So there was a stomach ache, no? Yan, that's the chief complaint, no, of the patient. Yan. And according to the patient, no, patient medical history, no, the patient was actually rushed to the ER, no, on a separate occasion due to myocardial infarction, no, yan. And then he was given this OTC drug, no, as parang way, no, to minimize another event of myocardial infarction. No? So she continued taking this drug, no, until she felt the stomach ache, no, which again led her to go to the hospital. All right. So number 18, no, which lipid could, which synthesis, or synthesis of which lipid could have been disrupted by discontinuous intake of this OTC drug. No? Ayan. So probably, no, let us first identify what is the OTC drug. Okay, yan. So yun siguro yung una nating sagutin. Sino kaya yung binigay na OTC drug for the patient? And according to her, it was because of her myocardial infarction. Okay? So normally, no, aspirin, okay, is given to patients. Yan. Aspirin is given to patients with myocardial infarction or yung even those patients na mayroong mga clot na yan, which causes yung heart to malfunction, right? Yan, kasi nga, the aspirin is a blood thinner, no? Ayan, so, it's a platelet aggregation inhibitor, no? Ayan, kaya siya binibigay for myocardial infarction, okay? So, that's the aspirin. Now, aspirin, no, in low doses, yun yung talagang purpose niya. At a higher dose, no, your aspirin is also an inflammatory drug, no, anti-inflammatory drug. It is also an NSAID, no, a non-steroidal anti-inflammatory drug. All right. Now, your aspirin is an example of a non-selective COX inhibitor. Okay, and your because your aspirin, okay, your aspirin is a non-selective COX inhibitor. Okay, it inhibits both prostaglandins and thromboxane. No, yan. So our answer here will be letter D. Both A and C are correct. Ayan. Number 19. Ayan. The drug is probably an inhibitor of which enzyme? Ayan. So nasabi na natin kanina, it's the cyclooxygenase, right? Ayan. And then let's proceed to number 20. Based on the experience effect and the potential use of the drug in her myocardial infarction, which do you think was the drug given to the patient? So our answer will be letter A, aspirin. All right. And so atorvastatin is an HMG-CoA reductase inhibitor. Okay. Their silostazole is also a platelet aggregation inhibitor, no, but it acts on the platelets itself. No, it acts directly on the platelet. Okay. It doesn't have anti-COX activity. All right. And then letter D, orlistat, this is a lipase inhibitor, no, and this is used commonly to um, uh, reduce, no, yung triglycerides or cholesterol levels. Okay? Yeah, so anti-hyperlipidemic agent yung orlistat. All right? So our best answer will be letter A, aspirin. Okay? Ayan. So now we move on to the nucleic acid. Ayan. So, tapos na tayo sa lipid, no? So the next part will be nucleic acid. All right. So let's answer first number 21. So number 21, the structure above is, bali, this is for numbers 21 to 24. No, you study the structure below. And number 21, the structure is which one of the following compounds. Okay. So how do we analyze this kind of problem? Okay. So the first thing that you have to do is identify whether you have a nucleoside, a nucleotide, or a base. Okay. So yun yung unang iisipin ninyo, no? So, meron ba akong nucleoside or nucleoside? Yan. So remember, when you say nucleoside, no, this is just sugar, which can be ribose or deoxyribose, and a base. And then pag sinabing nucleotide, this is sugar, phosphate, and a base. Ayan. Okay, sugar, phosphate, and a base. Okay, that's your nucleotide. Now let's look at the structure shown on the right. And so you have here the phosphate group, you have here the sugar, and you have here the base. So this is a nucleotide. Okay? Now remember, no, na in naming your nucleosides and your nucleotides, magkaiba yon. Okay? So for nucleosides, okay, remember na osine. No? Yan. So osine is the suffix, no, of your purines. No? So for example, adenosine, deoxyadenosine, guanosine, deoxyguanosine. Okay? Now for pyrimidines, no, yeah, for pyrimidines, it's idine. Yeah, so for example, you have your cytidine, deoxycytidine, uridine, and thymidine. Okay, thymidine is also your deoxythymidine. All right. Now, for nucleotides, no, the suffix will be ilic acid or elate. Yan, so elate or ilic acid. Yan, pareho lang yun. So ilic, medyo mabagal yung pick up ng aking device. Okay, so elate or ilic acid. And then, for purines yan, no? So for example, adenylate, adenylate, guanylate, deoxyguanylate. All right? For the pyrimidines, no, sabi ko last time, lagyan lagi ng ID, no? Ayan, para alam nating pyrimidine, no? Ayan, so idylate. So idylate or idylic acid, no? So idylic acid. Ayan, medyo mabagal yung pick up. Okay. Yeah, so idylate or idylic acid. All right? So that's for your nucleotide. All right? Okay. Now, let us first identify the type of sugar. Uy, nangyari. Yeah, so let us identify the type of sugar that is present in the molecule. Ito. Ay. Ayan. So, yung sugar na present ay isang galawin natin. Ayan. Yung sugar na present ay isang and this is an example of a deoxyribose kasi wala tayong sugar at that position. Okay, so that's the 2' position and there is no OH hydroxyl group, no, in that position. So this is a deoxyribose sugar, no? So this is your deoxyribose. Yeah, so this is your deoxyribose. All right. Now, we have the phosphate, we have the base, we have the sugar. So this is an example of a nucleotide, okay? With a deoxyribose for its sugar. So let us look at the choices. No? Tingnan natin kung may ma-eliminate na tayo. Guanylate, okay, no. Guanylate, no. Thymidylate, idylate, no. Idylate, no. So lahat ng mga choices ay nucleotides. Okay? So now, let us remove those that contain ribose instead of deoxyribose. Okay? Kasi sabi nga natin, the sugar that is present in the molecule is deoxyribose. Okay? So, of all this, no, ang pwede nating ma-eliminate na ay si adenylate, no? Kasi the fact that it is just adenylate means its sugar is ribose. Okay? Yan. Letter D, uridylate. Uracil can only be found in ribonucleic acid, so its sugar is always, no, ribose. Okay, so this is also eliminated. So there are only two. no, in the choices. Okay, that may be the correct answer. Okay. Now remember what I mentioned before, no, regarding the nucleic acid bases. Okay. So sabi natin, adenine and guanine are pure and uncut. Okay. So adenine and guanine are yan, pure and uncut. Yan. So anong ibig sabihin nito? Your adenine and guanine are purines. Okay? They are uncut. They are not cytosine, uracil, and thymine. Okay? So, ibig sabihin, your adenine and guanine have a large molecule. So, kasi yung purine molecule is big. Okay? Yan, di ba, drinawing natin yung purine molecule sa inyo last time. No? So, ito yung ating purine molecule. Yan. We have N here, we have N here. Tapos meron tayong double bond dito, double bond dyan. Nitrogen dito, nitrogen dyan. Double bond, double bond. Okay, so this is your purine molecule. Siyempre may H5. Yan. So that's your purine. Okay? Now, is this a purine or a pyrimidine? Yan. So based on the structure, okay, dahil maliit lang naman yung ating molecule, 1, 2, 3, 4, 5, 6, there are only 6 members in the heterocyclic ring. So this is a pyrimidine. Okay? So hindi siya purine, so eliminated na si letter B. Okay? So our answer is only letter C, thymidylate. Okay? Ayan. So remember that when you have a small molecule, okay, it's pyrimidine. Mas mahaba yung pangalan. Pag mas malaki yung molecule, that's purine. Mas maiksi yung pangalan. Okay? Yeah. So the basic structure of your um of your naghang yung utak ko. The basic structure of your pyrimidine is this. Okay? So pag ganito siya. 1, 2, 3. Yan. Okay? Yan. So tapos na yan. This is your pyrimidine basic structure, and then this is your purine basic structure. Alright? So pyrimidine, and then this is your purine. Ayaw sumulat. Parang Japanese yung sulat to. Okay. So, and another thing, no, that you have to remember, since this has a methyl group, no, that's your thymine. Okay, yan. So, yan na yung pinakamalaking clue. No? So, you have a pyrimidine with a methyl group, no, that's your thymine. Okay, and remember na, even if your thymine, no, even if your thymine is attached to the deoxyribose, we can drop the term deoxyribose. No? So, thymidine or thymidylate, no, and deoxythymidylate are the same. Okay? So the answer for number 21 is letter C. All right, number 22. This can be found in which nucleic acid? So ang ibig sabihin, no, yung structure at above, no, can be found in which amino acids? So I have repeatedly mentioned that thymine, letter T, can only be found in your DNA, no? Kasi it's always deoxyribose. Ayan, so letter A, DNA only. Number 23. In the nucleic acid, the structure above forms how many intermolecular hydrogen bonds with the base, no? With which base? Okay, so first let's identify which base, no, will thymine form a bond. No? So remember, according to Erwin Chargaff, adenine forms a pair with your thymine, AT, ano, and then si guanine forms a pair with your cytosine, no? So ATGC, no? Ayan. So yan yung tatandaan yung base pairing. Okay? So in other words, ang tama na dito sa mga choices ay yung mayroong A, no? Kasi thymine yung ating molecule doon. So it's obvious, no, that the answer is letter A. Ayan. So 2 and 8. No? So remember na between your adenine and your thymine, there are two hydrogen bonds. Between your guanine and cytosine, there are 3 hydrogen bonds. Okay? Ayan. So the answer is letter A. No? So the structure above your thymidylate forms two intermolecular hydrogen bonds with adenine base. All right. Number 24. The sugar in the structure above is mainly derived from which metabolic pathway? Yeah. So all you have to do is to count. No? Yan. How many carbons are present in this particular sugar? Yan. So bilangin natin. 1, 2, 3, 4, and 5. There are 5 carbons. So this is derived in letter C, pentose phosphate pathway. Right? So always remember that the pentose phosphate pathway has only two functions. No? It helps in the production of your nucleotides. Kasi your ribose is produced from the pentose phosphate pathway. Right? And it also does not produce ATP nor consume ATP. All right? Yan. So that's for number 24. But we're not yet done with the nucleotides and nucleic acids, no? So for numbers 25 to 26, consider the structure below. Ayan. So medyo din naman ako yung question on this, no? Ayan. Yung identification of the structures kasi sayang naman, no? Yan, kung inaral nyo ito kasi medyo nagtagal tayo dito last time. All right? So number 25, the structure above is which one of the following compounds? Ayan. So let's again do yung ginawa natin kanina. And so let's identify the parts. And so you have here a sugar, okay? You have here a base. Okay. Yan. Now the sugar that is present is a ribose kasi at position number 2, it has the hydroxyl group. So this one is a ribose. Okay? And dahil ribose siya, eliminate na natin lahat ng mga choices na deoxyribose yung nakalagay. So deoxy, deoxy. Dalawa na lang. Okay? Yeah, so para tong who wants to be a millionaire. Okay? We have already eliminated two choices. So dalawa na lang yung pagpipilian natin. Now, let's look at the base. No? Ayan. So is the base small or is the base big? No? The base is small, no? And it has the same structure like this one. Okay? Ayan. So, meron tayong ganitong structure. Okay? Ayan. So, kung titingnan ninyo, yan yun. Okay? So, this is a pyrimidine. Ayan. So, this is a pyrimidine. Pyrimidine. Ayan. So, dahil isa siyang pyrimidine, okay? Dahil isa siyang pyrimidine, okay? Pagpipilian lang natin ay si C, U, and T. No? So, cytosine, uracil, and thymine. Okay? Ayan. So, yung tatlo na lang, no? Yung pagpipilian natin. So, we have cytidine and thymine. Oy, pareho. No? Ayan. So, pareho tayong merong C at saka T. Okay? So, para hindi pa natin eliminate. But let's look at the suffix. Okay? So, sabi natin, kapag meron akong nucleoside, okay, nucleoside, ito yon, okay? If I have a nucleoside and I have a pyrimidine, the suffix will be idine. Okay, idine. Okay? Ayan. So, balikan natin ito. Ito ay idine, ito ay in. Okay? So, we eliminate letter D. The answer is letter A. Ayan. Okay? So, the answer is cytidine. Okay? So, letter D, thymine is the base. Ayan, wala siyang sugar component. Okay, so it's just the base without a sugar component. Okay? Now, what did you notice? No, ayan. So sana may napansin kayo in the way that I rationalize these set of questions. No? Even if you don't memorize the structure, di ba yan? Lagi ko sinasabi sa inyo last time, di ba? Do not memorize, just be familiar. No? Just familiarize yourself with the structure. If you are familiar with the structure, you don't even need to memorize the structure. Okay? You can just eliminate what needs to be eliminated. Okay? Tapos, for example, ako mismo, ako tanungin mo ko, draw out of the thin air certain molecules, I cannot, memorize them that easily, and unless ulit-ulit ko siyang dinodrawing. Okay? Yeah, but, most likely, you can eliminate the wrong answer. Okay? Yeah, so tulad ng ginawa natin, no? So the style, no, na ginawa ko, hindi ko pinakita sa inyo yung actual structures, no? Ngayon, no, hindi ko sa inyo pinakita yung actual structures ng cytosine, ng uracil, at saka ng thymine. Okay? So pinakita ko dito ngayon, no, in our rationale, without knowing those structures, for as long as you know the basics, the foundations, no, you can actually get your answer. Right? And but if you want to check, no, yung mga structures nila, of course, no, you can go back to your handouts, no, and you will see, no, na yung cytosine siya yung mayroong amine and carbonyl group, no. So ang kwento kasi niya last time na nakakatawa, no, para hindi makalimutan, si uracil at saka si thymine, yan yung para siyang nakaganon, no, na tao with two carbonyl functional groups. All right? Yan. So instead of having an amine, no, in this portion dito sa part na ito, ang carbonyl functional group. All right? For uracil and for your thymine. All right? Yan. So yan yung essence of these numbers. No? I'm just trying to show you, no, another way of learning, no, to answer problems or questions with structures in them. Okay? Number 26. The nitrogenous base in this structure is ayan. So nasabi na natin, no, it's a pyrimidine. Okay? Because it's a large molecule. Okay? So dalawa lang naman yung tamang sagot dyan, no? So your purine or si pyrimidine. Ayan. So letter D, pyrimidine. Number 27. Which one or which of the following is not, ayan, not a ribonucleoside? Okay? Ayan, so balikan natin yung sinulat ko dun sa itaas, okay? Pag nucleoside, okay, the ending for purines, no? The ending for purines ay osine. So adenosine, guanosine, deoxyadenosine, deoxyguanosine. Okay? Kapag nucleoside, no, ng inyong pyrimidines, no, idines. For example, cytidine, uridine, thymidine, deoxycytidine. Okay? And so yan yung mga nucleoside. So tingnan natin kung sino yung pwede eliminate. Yan. So dapat daw osine or idine. Uy, parang wala ako ma-eliminate. Okay? Yan. Parang wala, no? Parang impossible to answer yung question. Uy, may mga umalis pa rin. Okay. So yung next na gagawin natin is tingnan natin kung tama. No? Kasi osines only for purines. And remember that purines are adenine and guanine, nothing more. Okay? Your cytosine, uracil, and thymine are all pyrimidines. Okay? So ito ay pyrimidine, ito ay pyrimidine. Okay? And for pyrimidines, no, the suffix of your nucleoside ay idine. So this one is correct. No? This is a nucleoside, but cytosine is not. Kasi cytosine is just a base. Yan, okay? Yan, so it is not a ribonucleoside. It is just a base. Letter B, cytosine. Yan, okay? So 59. And nagsimula tayo mga around 70. Yan. Baka na perfect nila. So hayaan mo sila. Okay, number 28, and blank stated that DNA from any cell of all organisms should have a 1:1 ratio of pyrimidine and purine bases. So the answer is letter A, Irwin-Shorgaf. Okay, yeah, so remember that the base pairing was actually proposed, no, by Irwin-Shorgaf. So according to him, when he tested, no, the duplex DNA, ibig sabihin double-stranded DNA, he was able to note, no, that the adenines, the percentage of adenines, no, in the duplex DNA ay similar, no, very similar to the percentage of thymines, no, in the sample. And same is true for your guanines, same din yung kanyang percentage with your cytosine. No? So he proposed that there is a 1:1 ratio between adenine, which is a purine, and thymine, which is a pyrimidine, as well as guanine and cytosine. All right. So number 29, the scientists who performed detailed study on the structure of the DNA by X-ray crystallography were. So remember the note here is that, no, the scientists who performed detailed study on the structure of the DNA by X-ray crystallography, it's none other than the female scientist, Rosalind Franklin. Okay? And so do not ever forget that. No? And Rosalind Franklin is the one who worked hard, no, to produce the photo 51 or the photograph 51, okay, which I showed you in your quiz, okay? So yun yung photo 51. And it was Rosalind Franklin who actually initially proposed the sugar phosphate backbone, no, of the DNA. No? And surprisingly, what Rosalind Franklin was able to photograph, no, through X-ray crystallography, IB form of the DNA. Okay? But it was Francis Crick and James Watson who published, no, the DNA structure. Ayan. So ano na yan, canceled na yung dalawang yan. Okay, number 30. Which of the following is true about double helical DNA? Ayan. So let's look at the choices. No? Let's begin with letter D. Although they are associated in an anti-parallel fashion, the two chains have an identical base sequence. All right? So remember that when you say anti-parallel fashion, ibig sabihin nun, when one strand is in 5' to 3', the other strand is 3', 5'. Okay, yan. So they are anti-parallel. Okay? Now, remember that we cannot assume that both strands would have identical base sequence. Okay, yan. In fact, very rare. Yan. Na identical base sequence yung dalawa. Okay? Ayan yung dalawang DNA. Okay? Ayan. So, that being said, letter D is wrong. Ayan. Letter C. Unless the DNA is circular, the 3' hydroxyl groups of each chain are at opposite ends of the molecule. Ayan. Unless the DNA is circular, the 3' hydroxyl of each chain are at opposite ends of the molecule. Ayan. So, Ayan. So for this one, what is a circular DNA? No? So yung circular DNA ay yung plasmid. No? Ayan. So example natin dyan ay si plasmid DNA. Alright? Ayan. So yung plasmid DNA no, doesn't have ends. Okay? Ayan. So wala silang ends. Tapos, ayan. So they are circular. So ibig sabihin wala siyang end, no? Ayan. So this is actually true. Ayan. Okay? Ayan. So napaisip ako, no, na mali na naman ako ng orientation. And dapat sa E tayo nagsimula. Ayan. Okay. So, pero tingnan pa rin natin yung two other choices. Letter B, the chains have a backbone of linked glycosides. No, ayan. So remember ito, ang nagpamali dyan. The backbone is phosphate sugar. No, ayan. So this is wrong. Letter A, and the planes of the bases lie parallel to the helical axis. So remember, ito yung helical axis. The bases lie perpendicular, no, to the helical axis. So letter A is wrong. So our answer is letter C. No? Unless the DNA is circular, the 3' hydroxyl groups are at opposite ends of the molecule because the molecule is anti-parallel. Okay? Yeah. So the arrangement of the two strands is anti-parallel. All right? Number 31. The denaturation temperature of, or the melting temperature of DNA is, teka, magbura tayo, no? Ano to? Ayan. So for this number, ayan, dinefine natin to before, no? Yung melting temperature for the DNA. Let's begin with letter D. And the temperature in which the sugar phosphate backbone is completely hydrolyzed. Ayan. Remember that there is no hydrolysis of the phosphodiester bonds. Okay? In your DNA, no? When you denature the DNA. So letter D is wrong. Letter C, the temperature in which the N-glycosidic linkages are completely hydrolyzed. No? Now, you may wonder, nasan yung N-glycosidic linkage? Ito yun. no? And so the N-glycosidic linkage ay ito. Ito. Yan, okay? So that's the N-glycosidic linkage. No? Ganun din dito. Yan, meron din yan. N-glycosidic linkage. Okay? The one that connects the base and the sugar. Okay? So now remember na the N-glycosidic linkage is also not hydrolyzed, okay, by denaturation of the DNA. So letter C is also wrong. Okay? Letter B, yan, dalawa na lang. The temperature in which the entire length of the DNA is single-stranded. And letter A, the temperature in which 50% of the length of the DNA is single-stranded. Okay? Now, I mentioned no, in the review no, na it's difficult to actually determine the temperature where the two strands are completely separated, no? Where the hydrogen bonds no, in the two strands are completely removed, no? So letter B is wrong. Okay? So we always determine the denaturation temperature at the point where only 50% of the entire length is single-stranded. No? So ibig sabihin, only hydrogen bonds no, are broken in melting or denaturation of the DNA. All right? So that is for number 31. Okay, next number. Number 32. If the base sequence of a segment of DNA is CAG, which of the following sequences is complementary to it? Okay? Now, the way that we will do this no, is this. Remember na, pag 3' yan, dapat ang katapat niya 5'. And then pag 5' to, ang katapat niya ay 3'. Okay? And then, next na gagawin natin is, palitan natin. Ayan. So, we now write the complementary strand. Okay? So, ang complementary strand ng CIG, okay? Ang complementary strand complementary base of A is T. The complementary base of G is T. T is A. T is another A. Okay? And then A is T. Then G is T. T is G. Okay? Ayan. So that's our complementary strand. However, remember that we have to flip the strand, no? Kasi the way that the nucleic acids no would be presented to you will always be 5' to 3'. In fact, mabait pa ako dito, no? Ayan, sinulat ko yung ends, 5', 3'. Ayan. Okay? By convention, no, you can actually drop these numbers. Okay? Kasi when you see a DNA sequence, it's always 5' to 3'. Alright? So kung meron kayong dapat tandaan na hindi nyo na makakalimutan forever, no? So remember that we write nucleic acids 5' to 3'. Alright? Ayan. So babalik na rin natin lahat. So we have G, C, T. Ayan, sulat natin yon. We have 5' to 3', G, C, T. Ayan. Tapos patras, A, T, and then T, G. Ayan, 5' to 3' na yan. Okay? So, sino yung pinakakamukha niya? That's letter A. Ayan. GCT, AAC, EG. Okay, 5' to 3'. Alright? So, that is our answer no, for number 32. Alright, number 33. Another thing pala that I would like to highlight here is that ang sabi kasi dito ay complementary DNA. Okay, yan, complementary DNA. Kasi pag tinanong ay mRNA, that's a different matter. Okay? Yan, so number 33, the form of the DNA in physiologic conditions is or are the A form, B form, C, Z form, or D both A and C. Yeah, so remember that the A form is present no, in its dehydrated form. No? Medyo chubby yung A form. Ayan. Medyo chubby yung A form. It is right-handed. No? Ayan. Tapos medyo mataba siya. Ganito yun. Pag ganito yung itsura ng A form, okay, of the DNA. Tapos yung Z form naman, it's quite extended. Pag ganito. Okay? Tapos pinakasakto lang yung inyong B form. Okay? Ayan. So, the A form occurs when the system, when the conditions are dehydrating or in the presence of alcohol. Okay? And A form yon. So that's not the form of the DNA in the physiologic condition. So letter D is already wrong. Okay? Now the Z form is the only left-handed DNA. Okay? And it is present in some of the CPG islands na tinatawag. Okay? So itong CPG islands kasi na ito, these are regions in the genome which are very tight. Ayan, because of a large number of C's and G's. No? Remember na yung C at saka yung G, C nagpo-form ng 3 hydrogen bonds with G. No? Ayan, so ibig sabihin, kapag marami kang C and G in a particular region, mahirap kang i-denature. Okay, yan. Kasi mayroon kang triple bonds between C and G. Alright? And so, these are found in regions where there is high regulation, no, of transcription. Okay? Yan. So, that's the Z form. Okay? So, that's also not the DNA in the physiologic condition. So, the answer is letter B. And so, letter B, B form. Okay? Yan. So, number 34. Which of the following is a palindromic sequence? Ayan. So remember, no, sabi natin kanina, last week, no, pag sinabing palindromic sequence, they are inverted complementary bases. Okay? So ibig sabihin, they are complementary, no, on opposite ends. Okay? Ayan. So yan yung ibig sabihin ng palindromes. Okay? Or kung isipin niyo, no, the two strands are identical, no? Kapag pinagbaliktad mo sila. Okay? Yan. So they are identical kapag pinagbaliktad mo sila. Okay? Yan. So yun yung ibig sabihin natin ng palindromic sequence. Okay? Yan. The two strands, no? The complementary strand of the palindromic of the sequence with a palindromic, sorry, ulitin natin yun. The complementary strand of the strand with a palindromic sequence is identical, no, to itself. Okay? Pakita ko mamaya kung paano nangyari yon. No? But just to easily remember it, they are inverted complementary strands. Okay? Ayan. So, yeah, that's it. So, the palindromic sequence is important in the formation of hairpins. No? Ayan. So, remember, in raw independent, no, raw, RHO, in raw independent termination of transcription, okay, a signal that is in raw independent termination of transcription, a signal that can be used is the presence of the hairpin. No? So, ang ibig sabihin nun, kapag may nakita ng hairpin, no, sa mRNA, no, ibig sabihin, hinto na yung transcription. Alright? Ayan. So, alright. Now, how do we know kung sino sa kanila yung may palindromic sequence? No? So, hanapin nyo dyan kung sino yung mayroong ka-pair sa kabilang side of the molecule. Okay? So, let's try letter C. Okay? So, si letter C, G, ka-complement niya si C. Okay? Tapos, si C, ka-complement niya si G. Si T, ka-complement niya si A. Si A, ka-complement niya si T. Uy, sakto. That's your palindromic sequence. So our answer is letter T. So, ayan. So GCTA, PAGT. Okay? Now remember what I mentioned kanina, no? The complementary strand of a strand with a palindromic sequence is identical to itself. Okay? So tingnan natin kung identical nga ba yung complementary strand niya. Okay? So let me write the structure here. Uy, dalawa na naman. Ayan. So, sulat natin yung strand muna na ito para hindi kayo ma-confuse. So, you have G, ayan, so nabilisan niya. G, C, D, A, T, A, G, and C. No? Ayan. So, this is 5 prime. This is 3 prime. Okay? Now, let me write its complementary strand, 3 prime, 5 prime. Okay? Ayan. So, sulat natin, G is C, C is G, T is A is T, T is A is T, G is E, C is G. Okay? Now, baliktarin natin ito. Ang ganito natin siya basahin. Okay? This now becomes G, C, T, and G, C, T, A, T, A, G, T. 5 prime, 3 prime. Okay? Now, let's look at the two. No? Yan, tingnan mo to. At saka tingnan mo to. They are identical. Okay? Ayan, so they are palindromic. Or this one, only they are. This one is a palindromic sequence. No? Kasi its complement, no? Strand, so its complementary strand is identical to itself. Alright? Ayan. So kaya nga kanina, no, doon sa isang question, ayan, letter D is wrong. Kasi only strands which are palindromic would be identical, no? Ayan, identical sequences. Okay? Ayan. So hindi lahat. Alright? So clear ba 'yon? Ayan. So ano muna, sanity check tayo, sanity check. Ayan, so are we still okay? So can I see a thumbs up kung ano pa, nakakahinga pa? Okay, so may nakakahinga pa naman. Okay. Ayan. So, ayan, thank you for your response. Ayan, so kasi wala akong nakikitang questions, no? I am checking kasi even your chat, no? Kung ano. Ang napapansin ko, maraming umaalis. Ayan. Ayan. Okay. So next would be number 35. Which of the following are true about the eubacterial DNA polymerase 1? Ayan. So, tandaan, no? The DNA polymerase 1 and 3 are polymerases for prokaryotic, no? Ayan, prokaryotic replication. Okay? Um, they are easy kasi, no? They are easy to evaluate, no? Ayan. So, madaling na-discover yung DNA polymerase 1 and 3, no? In bacterial replication. For the eukaryotic replication, it's quite complicated. Okay, and so in fact, no, what happened there, no, was that very very late, no, na discover yung mga DNA polymerases for eukaryotic replication. Okay, and then para lang mas madali ninyo ma-identify, pag numbers, prokaryotes yon. Pag Greek alphabets, alpha, beta, gamma, delta, epsilon, those are eukaryotic DNA polymerases. All right. Ayan. So number 35, which of the following are true? So ibig sabihin more than one yung answer about the eubacterial DNA polymerase 1. Ayan, number 1. Simulan na natin sa number 1. It is the main polymerase that continuously adds bases from the RNA primer. Okay? So now remember that the start of the replication, no? Sige, drawing tayo for that. Whiteboard tayo. Ayan. Remember that the start of the replication begins with the binding, no, of your pre-priming complex. Okay, yan. So yun yung nauna. Yan. So magdodrawing muna ako ng DNA. So ito yung template strand. Tapos ito yung other strand. Okay? So this is your 3 prime, no? And then this is your 5 prime. Okay? So para mas masaya, ibahin natin yung kulay nung nasa baba. Hmm. Ayan. So this one is 5 prime and this one is 3 prime. Okay? Ayan. So remember that the first thing that happens during replication is the binding of the pre-priming complex. Okay? And it requires 3 things. Okay? So you have an RNA primer, or you have the primate, you have the primate, okay? Which produces the RNA primer. Ayan, so kay primase ito. Tapos you have the DNA helicase. The DNA helicase which unwinds, no, or which breaks the hydrogen bonds, no, in between the two strands of the DNA. And then you have number three, you have your single-stranded DNA binding protein. Now the primase, no, adds a primer to the template strand, no, also known as the also known as your leading strand. Okay, yeah. So this one is your leading strand, and the other one is your lagging strand. Remember that DNA polymerase always adds bases 5' to 3'. Okay? So, kunwari, no, yung inyong DNA polymerase, kunwari ito siya. Ayan. So, si DNA polymerase, ito siya. Magdadagdag siya ngayon ng bases pag ganito. Ayan, pag ganito. In the leading strand. Kasi nga, the leading strand should have 5' to 3'. Andito yung 3' niyan. Okay? For the lagging strand, it's quite different, no? So ito si DNA polymerase, ayan, so ito ulit siya, may malaking molecule yun. So nandun siya sa unwound ng DNA, si primase, no, maglalagay siya ng primer. Ayan, so andito yung primer. Ayan. Pero instead na mag-add siya, no, forward, paatras yung pagdagdag niya. So pag ganito, pag ganyan yung pagdagdag, no, ng inyong DNA polymerase in the lagging strand, no? So this is your 5', that's your 3'. Okay? So sana na-imagine nyo yun, no? Yung kulay red, yung red na red, this is your RNA primer, and this is your RNA primer. Tapos this is the newly synthesized DNA, and pag ganito. Pag ganito to. Okay? And then gagalaw ngayon yung inyong DNA polymerase, yan, gagalaw siya. For the leading strand, wala tayong problema, dire-diretso lang yan. Okay? For the lagging strand, no, kung nasaan yung DNA polymerase mo, doon kakabit yung RNA, yung inyong RNA primer, and then synthesize siya ng DNA patras. Okay? Gagalaw ulit yung inyong DNA polymerase. Okay? Of course, while that happens, yung inyong pre-priming complex, no, ito yung inyong helicase, no? Tapos nandito rin yung inyong yung inyong single-stranded DNA binding protein. Okay? They unwind, no? They unwind the DNA. All right? Yan. So you're going back to this drawing, pag gumalaw yung inyong DNA polymerase, ito, okay? For the leading strand, wala siyang problema, hindi siya magdadagdag ng isa pang RNA primer, okay? It will just add bases, no? Ayan, in 5' to 3' direction. But for the lagging strand, maglalagay pa rin siya ng isang RNA primer, and then magsisynthesize siya ng or maglalagay siya ng complementary strands 5' to 3', like that. Okay? And because of that, we call these, no, dahil may mga breaks dito, may break din dito, ayan, we call these your Okazaki fragments. Okay, yeah. So those are your Okazaki fragments. Okay? Now, take note, no, na, ano ang sasabihin ko? Take note that the main DNA polymerase, no, that adds, no, nucleotides to the growing chain, no, of the DNA would be your DNA polymerase 3. Ayan. So yung identity ng polymerase na ito, it's your DNA polymerase 3. Okay? Ayan. So balik lang tayo muna doon sa mga choices. Ayan. So that's DNA polymerase 3. Ang tanong ay DNA polymerase 1. Okay? So number 1 is wrong. Okay? It is not the main polymerase that continuously adds bases from the RNA primer. Okay? So tingnan natin number 2. It has both 5' to 3' exonuclease activity, no? And 3' to 5' exonuclease activity. Now, what do we mean by these two terminologies? Okay, ito. 5' to 3' exonuclease activity and 3' to 5' exonuclease activity. So, balik tayo dun sa drawing ko kanina. Tanggal ko yung chat. Okay. Okay. So, now, once the two strands, no, the daughter strands are produced, no, yung leading strand at saka yung inyong lagging strand, the problem here is that meron pa rin tayong kailangan tanggalin. No? We have here the RNA primer and we have here the RNA primer. Ayan, di ba? Yan, kapag nakompleto ng ma-synthesize yung inyong daughter strands, so let's say na-complete ko na to, yan, meron tayong RNA primer. These are not DNA. No? So, ibig sabihin at the start, no, or at the end of replication, you have a hybrid DNA. Meron kang RNA, meron kang DNA. Okay? So, you need to remove, no? You need to remove the RNA, no? The RNA primers. Okay? Now, dito papasok si DNA polymerase 1. Okay? Yeah, so si DNA polymerase 1, and let me erase this one. Ayan. So, let me add color. Ayan. Ay, sorry. Ayan. So, papasok ngayon si DNA polymerase 1. Anong kulay gagamitin natin? Ito, no? Si DNA polymerase 1. Ayan. So, DNA polymerase 1. 1. Ang gagawin ni DNA polymerase 1 is tatanggalin niya ngayon yung RNA primers. Okay? Yeah, so para matanggal niya yung RNA primers, kailangan niyang i-cleave ito. Okay? At i-cleave niya ito. Okay? So same with this one, tatanggalin niya dito, tatanggalin niya dito. Remember that ito. Okay? Yeah, so remember that this is 5 prime. Okay? This is 3 prime. This is 5 prime, this is 3 prime. So in other words, that activity is called your 5 prime to 3 prime exonuclease activity. Ayan. So this is an activity where oligonucleotides can be removed, no? From 5 prime to 3 prime direction. Okay? Ayan. So yan yung DNA polymerase 1. Okay? Now, aside from that, no, aside from that property, may iba pang ginagawa yung inyong DNA polymerase 1. So, natanggal na ngayon yung RNA primer. Yan, so, burahin natin yung mga to. Yan, natanggal na yung RNA primer. Yan. So, dahil natanggal na yung RNA primers, ano yung kailangan gawin? Kailangan nating dagdagan ng DNA. No? So, papalitan natin ngayon yung RNA primer, lalagyan natin siya ng DNA. Yan, pag ganun. Okay? So instead na RNA, yung ilalagay, bali syempre, pag nandyan yung DNA polymerase mo, ito muna yung gagawin niya. Okay? And then, lilipat yung inyong DNA polymerase dito. Ayan. So, magdadagdag sa ngayon ng RNA, ay ng DNA dito. Okay? Tapos, ilipit yung inyong DNA polymerase dito. Okay? It will add DNA here. Okay? Kaya, papalitan niya yung RNA. No? That is what you call the 3 prime, yung ginawa niya, yung synthesis of the or adding of the bases, that is called the 5 prime to 3 prime polymerase activity. Okay? Ngayon, an important feature of the DNA polymerases is the proofreading, no, property. So, ibig sabihin, let's say, nagdadagdag siya ng bases dito, yan, 5 prime to 3 prime, na-realize niya, uy, may mali akong nilagay dito. Ayan. Okay? So, what will happen is, tatanggalin niya ngayon yan, aatras siya, okay? Buburahin niya yan, okay? And then, uulitin niya yung kanyang pag-synthesize. Okay? Yan, ulitin niya para tama na. Ay, sorry. Ayan. Ano ba yan? Ganun. Alright? So, yung pag-remove niya, that is the 3 prime to 5 prime exonuclease activity. Okay? Remember that the 3 prime to 5 prime exonuclease activity is the proofreading property of your DNA polymerase. So in other words, DNA polymerase 1 has both 5' to 3' exonuclease activity to remove the primers, the RNA primers, and 3' to 5' exonuclease activity to remove wrongly incorporated bases. No? Ayan. So let's go back to the problem at hand. Ayan. So letter number 2 is correct. Okay, so your DNA polymerase 1 has both these activities. All right, so tingnan natin number 3. It fills the gap between Okazaki fragments in the lagging strand during replication. Okay, balikan natin yung drawing ko kanina. Ayan. So while, ayan, so maraming nalilito kasi dito. While the DNA polymerase is able to add DNA no, in place of RNA primers, no, ito yon, ayan, ito yung mga dinagdag niya, no, ito. These are not yet complete. No? May mga breaks yan. So yung mga breaks nila ay nandito. Ayan, nandyan, nandito. Okay? Tapos nandito. Ayan. Okay? And even this, no? Ayan, mayroong mga breaks yan. Okay? May mga breaks dyan. Okay? So, what fills in this gap? Ayan. So, while kumpleto na, no, yung bases, ang problema, wala lang siyang phosphodiester bond. Okay? Yan. Kompleto yung bases, wala lang phosphodiester bonds. So, yung magko-connect sa mga fragments na ito, no? This is what you call the DNA ligase. Ayan, dyan papasok si DNA ligase. The DNA ligase will fill the gaps, no? Ayan, that are present, no, in your Okazaki fragments. Okay? Ayan, so let's go back doon sa ating drawing kanina. Ayan, so it fills the gaps between Okazaki fragments. This is not the function of your polymerase 1. It's the function of your ligase. Okay? Number 4, it removes the RNA primer and replaces them with complementary bases. This is correct. No? Ito yung in-explain ko kanina. It's because of the 5' to 3' exonuclease activity and 5' to 3' polymerase activity of your DNA polymerase 1. Okay? Number 5, it is highly error prone due to lacking proofreading property. Remember that your DNA polymerase 1 has 3' to 5' exonuclease activity. So meron siya nito. Okay? So this is wrong. So only 2 and 4 are correct. Polymerase 1. At parang walang sagot. 2 and 4. Ayan. So walang sagot. So sige, 2 and 4 is our correct answer. Ayan. Okay? Ayan. So, or unless, ano ba yung ibig sabihin ko sa number 3? Pero I think it's just 2 and 4. Okay? Ayan. So, remember, no, yung nagko-connect between the gaps that your ligase. Okay? Ayan. Okay. So, number 36. Now, which enzyme catalyzes the separation of double-stranded DNA into single strands to allow each strand to be copied during replication? Another name of this enzyme is DNA topoisomerase. Okay? Yan. And that enzyme is letter A. Yan. So, DNA gyrase. Okay? Yan. So, nasabi ko na halos lahat ng uses ng ibang mga choice kanina. Number 37. Which is the primary or the main prokaryotic DNA polymerase responsible in adding nucleotides to the RNA primer in the growing chain at the leading and lagging strands. No? Ayan. So, remember na what we mentioned as the main prokaryotic DNA polymerase ay si DNA polymerase 3. Alright? Okay, next number 38. RNA-dependent DNA polymerase and is also known as. No, yan. So, remember when I presented the central dogma, and you have the replication, you have the transcription, and the translation. Now, in some organisms, or not really organisms, in some particles, let's say viruses, they have RNA only. No? So, yung RNA, no, sometimes need to be converted to DNA, okay, through an enzyme that is known as RNA-dependent DNA polymerase. Okay? And this enzyme is also known as letter B, reverse transcriptase. Okay? Yeah, so reverse transcriptase. Yan. Kasi The process of converting the DNA, the RNA into DNA is called reverse transcription. Okay? Yan. Now, kung nalito kayo dito, letter C, RNase, no? This is nucleosidase, no? This one cleaves, no? Cleaves the RNA, RNases, the RNA, no? The RNA molecules, okay? At kinakat into bases, no? Yung RNase, okay? That's the RNase. Ayan. Medyo na ang labo, no? Kasi RNAS, no? Ayan. And RNase, no? Enzyme that cleaves your ribonucleotides or ribonucleosides. Yan. And then, letter D, topoisomerase, this is also known as DNA gyrase. It relaxes, no, or it catalyzes the transformation of DNA conformations. No? Yan, yung tinatawag natin na mga topoisomers. Okay? Ito yung may supercoiling, negatively supercoiled, positively supercoiled, no? Or even relaxed. Ayan. So, topoisomerase yung kakatalyze nun. Ayan. So, may nagtanong kung yung bonus, yung number 35, talagang yun talaga. Sige, i-bonus natin, no? Kasi medyo confusing yung ibig sabihin ng number 3. No? Ayan. So, although, yung for number 3, yun. So, liwanagin natin, no? Ayan, para hindi kayo malito. Ayan. So, confusing kasi yung pagkaka-term doon sa number 3, no? Fills the gaps between Okazaki fragments, no? So, kung ang ibig sabihin natin ng fills the gaps, no, ay yung replacement of the bases where the RNAs are removed, okay? Of course, that's DNA polymerase 1, okay? So, ano yung ibig ko sabihin? Di ba kanina, ito ay mga RNA, okay? So, yung pagkaalis ng RNA, papalitan siya ng, na pagkaalis ng RNA, dami ko nabura. Pagkaalis ng RNA, drawing natin. Pagkaalis ng RNA, papalitan siya ng DNA by the DNA polymerase 1. Ayan. So, ito yon. And ako rin naman nagsulat ng question na yon. No? Bigla ko na-realize, medyo confusing siya. Ayan. So yung pag-replace, no, ng DNAs in the Okazaki fragments, when you remove the RNA, that is your DNA polymerase 1. Okay? But if we mean, no, na kino-connect yung mga fragments in the Okazaki fragments, okay? That is the function of your DNA ligase. All right? So siguro for number 35, yeah, we can make it a bonus for everyone. All right? Okay, so pero sana clear, no? Yeah, kasi it doesn't matter here, no, if kung ano ba, kung tama ka o mali doon sa question. What is important is you know why that is the correct answer or not. Okay? Ayan. So yun naman, yun yung mas importante dito. Okay? Ayan. So number 39. An adapter molecule which helps incorporate the correct amino acid on a growing polypeptide chain, no, is your, ayan. So the term that is important here is adapter. Okay? So adapter molecule, no? So pag sinabi nating adapter molecule, ibig sabihin it carries with it an amino acids, okay? And it has the anticodon to the codon. Okay? So that is the function of your letter D, tRNA. Okay? So para lang kilala niyo yung mga nakasulat dito na choices, mRNA, this is the messenger RNA, which is the transcript of the DNA that is important, no? In the synthesis of peptides, okay? And then rRNA is your ribosomal RNA. It is the RNA that is in complex with the protein in the ribosome, okay? Kaya siya tinawag na ribosomal RNA. And then the letter C, snRNA, this is your small nucleolar RNA, okay? Ayan. So it is inside the nucleus, particularly the nucleolus, okay? Yeah, number 40, which is true about codons. Ayan. Letter A is codons vary from one organism to another. This one is wrong. Ayan, kasi remember, codons are universal. Okay, yan. So, kinakalaban nito yung universality of the codon. and universality of the codon. Ayan. Okay. So next is letter B. Only one codon encodes for one amino acid. Okay? So only one codon encodes for one amino acid. Ito yung medyo confusing dyan. Only one codon encodes for one amino acid. No? Ano ibig sabihin nito? If you have one amino acid, iisa lang yung codon na nag-produce sa kanya. Okay? But is that true? Okay? Yeah, is that true? And that is not true, no? Kasi remember, no? Meron tayong tinatawag na degeneracy, no? Ayan, degeneracy of the codon. Degeneracy of the codon. Ayaw magsulat na matino. Okay, so degeneracy of the codon. So pag sinabi nating degeneracy, siguro lilipat na lang ako. Annotation na lang. Pag sinabi nating degeneracy of the codons, ibig sabihin, multiple codons actually encode for one amino acid. And this is to minimize the effects of mutation. No? Ayan. Kasi if there would be mutation, then kapag hindi degenerate yung ating codons, then a different amino acid will be incorporated in the growing peptide chain. No? And that will, that might be a problem. No? Ayan, that might be a problem. And so maaari siya mag-cause ng mutation. Oh, ayan. So, di ba meron tayong um, missense, nonsense, no? And silent mutation. Okay? Ayan. So, yun yung, ano, no? Yung different types ng mutation natin. So, pag sinabi nating nonsense mutation, okay? And nonsense, nahinto. No? Ayan, nahinto yung growing peptide chain because there's the stop codon. Okay? Pag sinabi naman natin na ano na silent mutation, there is no alteration in the protein product, no? Even if the DNA sequence has changed. Okay? And then pag sinabi naman nating missense mutation, there is a change na in the amino acid because of the mutation in your DNA. Okay? And we don't want that to happen. Okay? So letter B is wrong. Okay? Ayan. So letter C, one codon encodes for multiple amino acids. Ito kabaliktaran, no? Isa lang yung codon, pero multiple amino acids yung ini-encode niya. This is again wrong. Okay? Kasi remember that your codons are specific. No? Ayan. Meron naman tayong tinatawag na specificity of codons. Okay? So pag sinabi niyo specific yung codons, one codon, no? Only encodes one amino acid. Okay? Ayan. However, there can be letter D, multiple codons that will encode the same amino acid. No? Ayan ulit yung degeneracy of the codon. Alright? So, clear ba tayo doon? So, for number 40, the answer is letter, ayan. Aha, ayun na yung magiging problem. Ah, number 40, letter D. Multiple codons encode for a single amino acid. Okay? Ayan. So number 41. Which amino acid is incorporated by the start codon? Ayan. So first, no, sino ba si start codon? Your start codon is your A, U, G. Ayan. A, U, G. Okay? So AUG encodes for which amino acid. No? So if you look at the codon table, no, this one is actually letter C, methionine. Okay, and remember, no, before peptide synthesis occurs, no, methionine is first activated. Okay, and so it is converted into formyl methionine. Okay, and so number 41. Which codon signals the stop of peptide elongation? Ayan. So the stop codons, no? The stop codons ay sino? Sila UGA, UAG, and UAA. Kasi UAA. Ayan. Okay, medyo mabagal yung pick up. Ayan. So UGA, UAG, UAA, no? Ayan. So yan yung ating stop codons. So sino yung lumabas sa choices? We have UAA, no? Ayan. So letter D. Okay? And then number 43. Ayan, so medyo magbura tayo. Clear all. Ito lang yung problem sa annotation. Hindi siya sumasama. Okay. So for number 43, which technique is used in the in vitro amplification of nucleotides? Ayan. So in vitro, ang key term here, no? Ayan, in vitro. Ayan. So letter A, agarose gel electrophoresis. Remember, agarose gel electrophoresis is just a method of evaluating, no, DNA. So that is not the correct answer. Letter D muna tayo. Restriction enzyme digestion. And restriction enzyme digestion is needed, no, to transfer a gene to a plasmid. Okay? Yan. So that is also not the correct answer. So dalawa na lang yung choices natin. Letter B, molecular cloning. Or letter C, polymerase chain reaction. Remember that in vitro, amplification is PCR, polymerase chain reaction. Okay? It resembles, no, the steps in your replication. You have the, di ba, steps in replication, you have the denaturation, no, the annealing of the RNA primer to the DNA template, and then elongation. Yun din yung steps in your PCR. So number one, you have the denaturation. Number two, you have the annealing of the primers. And number three, you have also elongation. Okay? So letter B, molecular cloning is not, no, in vitro amplification um, because you actually have your cells. Ayan, meron kang bacterial cells. Okay? And the bacterial cells amplify the DNA for you, no, while they grow in plates. Okay? So letter C is our correct answer. And so for number 44, blank is a laboratory analysis method used to study RNA. Oh, yeah. So remember yung kwento ko, no? Meron tayong northern, southern, and western blot. Okay? Eliminated na yung A, immunoblot. Although immunoblot is also common, this is your ELISA. This is ELISA, no? Ayan. ELISA. Ayan. ELISA is enzyme-linked immunosorbent assay, no? ELISA is a commonly a method of analysis of proteins, no? Ayan, ELISA yon. Ayan. Aside from ELISA, of course, we have Western blot. Okay, ayan. Western blot is also for proteins. Okay? And then, Southern blot is for DNA. Ayan. And then, Western blot is for protein. Ayan, ayaw na naman magsulat. Ayan, teka lang. Sige na nga, mag-annotate na lang tayo. Proteins. And then for northern blot, this is for RNA. Okay? So our answer is letter, ayan, so letter, ano tayo? Letter B, northern blot. Alright? Ayan, so let me again erase everything. Okay? Yan lang, no? Mag-erase ako na mag-erase. Because hindi gumagana yung aking, or masyado na raw maraming nakasulat. Ayan. Let me just erase it. Okay. Buhay natin para malino. Okay, next. Number 45. Ayan. So number 45. Ayan. The amination of adenine in the oligonucleotide GCTA-TAGC through the action of nitrous acid results in, ayan. So ano ba yung nangyayari, no, kapag tayo ay mayroong deamination of adenine? So remember, no, when adenine is deaminated, okay, it preferentially pairs with cytosine. Okay? Yan, so ulitin natin yon. And when adenine, no, is deaminated, okay, this one preferentially pairs up, no, with your cytosine. Okay? Ayan. So dahil nag-pair up, no, si adenine, okay, kay cytosine, okay? Sa susunod na round, okay, sa susunod na round ng replication, cytosine will pair up with guanine. Okay? So that being said, the originally AT, no, ayan, becomes GC. Okay? where yung A becomes a G, no? And these are both purines. Ayan, so these are both purines. So, the type of so the type of mutation, no, the type of substitution that occurred, no, is a letter C transition. Okay, so the adenine became a guanine. Okay, and so there is a purine to purine, no, purine to purine mutation, and that is a transition. Okay, so remember na kapag sinabing transversion tayo. Pag sinabing transversion, purine becomes a pyrimidine and pyrimidine becomes a purine. Okay? Kaya pag sinabi namang deletion, an entire fragment or a base is removed, no? Insertion, may pinas. Okay? Kaya, so number 46. Which mutation of the sequence GAT-CCT is a transition? Okay? Kaya, so to be able to answer this question, no, tingnan natin kung ano-ano yung mga possibly mutated na mga um, bases. Okay? So, for letter A, so tingnan natin no, kung sino yung mutated sa kanya. Ayan. So, parehong 5 prime, ano to, it's the same sequence. Okay? Yan, hindi to yung complementary, no? So, GAT-CCT, ang nagbago sa letter A ay ito. Okay? So, the adenine, no, became guanine. Okay? And that type of mutation is a transition. Okay? Now, letter B. Tingnan natin yung letter B. GTT-CCT. Ayan. So, ganun pa rin si A naging T. Okay? Adenine is a purine. Thymine is a pyrimidine. So, this is an example of a transversion. Okay. Tapos, letter C, GTA, TCCT. No? GTA, and dalawa yun. Ayan. So, GTA, so, kulayan natin pareho. No? So, yung A niya naging T, yung T niya naging A. Yung T naging A. Okay? Tapos, okay, hindi pala. Yung TCC, teka lang, burahin muna natin. Ang nangyari is this, no? Yan, ito agad napansin nyo. This is your TCCT. Ayan, so this is your TCCT. Ayan. And ito yung TCCT. Ayan. So what happened really is, there is, no? Burahin ko din yung isa pang hinighlight ko. There is A, nasan si A? Ito si A. Ayan, ayan siya, no? Tapos si TCC ay ito, TCCT. So ito, kulayan na rin natin si G. Ito si G. Okay, so what happened here is that there is insertion. Okay? In-insert the letter T. Okay? Ayan. So for letter C, okay, it's not um it's not a transition, no? But it's an insertion. Okay? So there's insertion of T. Ayan. So the answer here is letter A, no? Ayan. Kasi nga adenine became a guanine. Okay? So possibly it was deaminated, no? Your adenine. Okay, so number 46. So for number 46, which is true, which of the following is true about glutamine? Ayan, nasa amino acid na tayo. Okay, glutamine, ayan, letter A, contains 3 titratable groups. That's not true, no? Kasi glutamine, let me draw the structure of glutamine. Yung glutamine, ganito yung structure niya. CO, minus, tapos T, lagay natin dito yung H, tapos NH2, NH3 plus, tapos 1, 2, 3. O, this is CH2, this is CH2, tapos this is N, NH2. Okay? Yeah, so this is your glutamine. Okay? So remember, it's an amide of glutamic acid. Glutamic acid has 3 carbons, no, in the R group. So 1, 2, and 3. Okay? Ayan. So The titratable groups are only two. Ayan, so ito lang yun, 1 and 2. Okay? This is not a titratable group. Okay, that's the amide, no? That's a non-polar group. So letter A is wrong. Okay? Letter B, it is an acidic amino acid, wrong. No? In fact, this is the amide of an acidic amino acid, so B is wrong. Letter C, contains an amide group. No? Ayan, this is correct. Ayan. Letter D has E as its one-letter symbol. Remember, for glutamine, the one-letter symbol is N. No? So letter D is wrong. So our answer is letter C. It contains an amide group, right? Yan. So now let's proceed to number 48. And so for number 48, which of the following amino acids contains an imidazole ring? Okay? Yan. So isa-isahin natin sila, no? Simulan natin kay letter D, tryptophan. So tryptophan contains an indole, no? Asa na? Tryptophan contains an indole ring. Okay? Your phenylalanine contains a phenyl ring, no? Or a benzene, no? A benzene ring. And then your lysine contains an aliphatic amine, and only histidine contains the imidazole ring. All right? And so our answer is letter A, histidine. Okay? Yeah, number 49. Yeah. Which amino acid gives a positive result in nitrous acid test? Remember that nitrous acid test is a test for amino acids with three amine. No? Ayan, with three amine groups. And there is only one amino acid with a free amine group, C, lysine. Letter B, lysine. All right. Number 50. Which gives a red colored, which gives a red colored solution with alpha naphthol and sodium hypobromite. Ayan. So alpha naphthol and sodium hypobromite. Ayan. So what do you remember about this test? No? So remember, inisa-isa natin itong mga test na ito. No? Sodium hypobromite or sodium hypochlorite is used in a test called Sakaguchi reaction or Sakaguchi test. And remember, sinabi ko last time, pag sinabing Sakaguchi, arginine. Ayan, Sakaguchi, argi, no? Ayan, arginine. So letter A, arginine. All right. Number 51. The test described in the previous item is known as letter B, Sakaguchi test. Ayan, so isa-isahin lang natin, no? So letter A, si Hopkins-Cole reaction. This is a test for tryptophan, no? And glyoxylic acid yung ginagamit na reagent for for Hopkins-Cole reaction. And then Snoop test is a test for histidine. Molish test is a test for sugars. Okay? Ayan. Number 52. Which test involves nitration of activated aromatic ring in amino acids? Ayan. So for this one, no, ang key term dito is nitration. Okay? Kasi among the choices, no, ayan, so tingnan muna natin yung A and B. Si A, Melon's test, this is a test for tyrosine. No? This one involves mercuric sulfate, no, and not nitrogen or not nitric acid. Okay, there's no nitration in Melon's test. Okay, so A is wrong. Letter B, nitrous acid test is a test for the preamines. No? So that is also wrong. Dalawa na lang to. The polysdiazo reaction and xanthoproteic test. Now remember, yung polysdiazo reaction involves the formation of an azo dye. Okay, yan. Through the coupling, no, of an aromatic diazonium salt with sulfonilic acid, with the aromatic amino acid. Okay, yung diazonium aromatic diazonium salt comes from your sulfonilic acid. Okay, so letter C is also wrong. So the only answer here is xanthoproteic test. And remember that xanthoproteic test involves nitric acid. Okay, it involves nitration. of the aromatic rings that have been activated. So particularly, ang nagpo-positive dito would be your tryptophan, no, and your tyrosine. Okay, number 53. All amino acids are chiral except ayan. So, ulit-ulit natin sinabi ito last time. Letter D, glycine. Yan, para mas maraming umalis, no? Okay. So, I'm wishing them luck. No? Sana, ano, perfect kayo. Okay. So, number 54. Which amino acid accelerates the formation of loops in proteins due to its flexibility? Ayan. So, flexible, no? Dahil nga, hindi sa chiral. That's letter D, glycine. Number 55. Which amino acid causes a kink in polypeptide? Parang bulto-bulto silang nawawala. Oh. Mga 10 mahigit nawala in a short span of time. And which amino acid causes a kink in the polypeptide and can be found in beta bands? Isa lang to, no? So this is your amino acid, no? Na amino acid, which is your proline, no? It's the only amino acid with a cyclic secondary amine group. Number 56, methionine, no, has a pK1 and pK2 value of 2.28 and 9.21 respectively. What will be its net charge at pH 8? Okay? Ayan. So, for this one, no, so let us first draw the structure of methionine. So, methionine, no, has this structure. So, COO minus CH, tapos N8, um, N8, sige, i-protonate muna natin lahat. I-protonate ko na rin. And tapos, CH2, CH2, S, CH3. Ayan. So, that's your methionine. Okay? Now, remember yung drawing natin before. Ayan. So, you have the 2 pK values, 2.28 and then 9.21. Okay? Now, the pH, no, the pH is pH 8. Yan. Now, remember what we mentioned last time, no, na this is your pK1, this is your pK2, and we always refer to the protons, no, in these functional groups. Okay? Sila yung mayroong pK values, no? Ibig sabihin kasi ng pK, actually ay pKa1, pKa2. Ibig sabihin acid dissociation constant, no? Now, for the, ayan, for pK1, which is 2.28, no, remember na kapag nasa left side, protonated. Kapag nasa right side, deprotonated. Okay? Ayan, so dahil deprotonated yung ating pK, yung ating proton, no, in the pK1, so tatanggalin natin siya. So this will now have a negative charge. Ayan. Okay? And then for pK2, yan, so bubuhayin natin ulit ito. For pK2, ito naman yung meron tayo. Over here, sorry. For pK2, and on the left side of it, ayan, you have protonated. So dahil nag-disappear yung pH. On the left is protonated, tapos on the right is deprotonated. Yan, okay? So dahil yung pH 8 ay less than 9.21, this one is protonated. Okay? So we keep that. We keep that. Okay? So this is -1, this is +1. So we have letter B, 0. Ayan. So letter B is 0. Ayan. Methionine at pH 8 will have a 0 net charge. Ayan. Number 57, histidine. Ayan. So for histidine, histidine has pK1, pK2, and pKr values equivalent to 1.82, 9.17, and 6. Which of the following statements is true about histidine? Ayan. So let's begin with letter A. Its isoelectric point is equal to 5.50. Ayan. Now, we have to take note kung anong klaseng amino acid ba si histidine. No? Ayan. So histidine is a basic amino acid. And because it is a basic amino acid, we will get the two higher pK values to determine its isoelectric point. Okay? So we just get yung average, no, of these two values. So 9.17 plus 6 divided by 2 is equal to 7.5. Okay? Ayan. So the correct answer here is 7.585 or 7.59. Okay? So A is wrong. Letter B. It has a net charge of -1 at physiological pH, which is 7.42. Okay? So the pH is 7.42. Okay? So remember na yung pH na yan is greater than the pK1. So ibig sabihin this one is deprotonated. So -1 yung charge ng ng carboxylic acid functional group. Now this is your pK2. No? It is on the left side, no, of your pH. The pH 7.42 is on the left side of this pK value. So ibig sabihin protonated. No? So the amine group is protonated. So the amine group will have a charge of +1. Okay? Now, pH 6, pH 6 is pH 6 is on the right side, no? Ayan, of the pH 7.59. Okay, hindi baliktad yun. The pH 7.42 is on the right side of pK6, okay, which is the aromatic, no, yung imidazole ring, no, proton of your histidine. Okay, so anong ibig sabihin nun? No? Ibig sabihin, the third group, no, the third ionizable group in histidine will be deprotonated. Okay, yeah, so deprotonated siya. And once deprotonated yon, no, wala siyang charge. Okay, yeah, so zero yon. Okay, deprotonated kasi. No? It's an amine. No? So yung amine, pag deprotonated, zero yung charge niya. Okay? Yan. So, -1 plus 1. No? So, the net charge is actually 0, not -1. Okay? Now, letter C. It is an acidic amino acid. Ayan. So, na-mention ko naman kanina, histidine is a basic amino acid. Ayan, together with lysine. No? Ayan, which is also a basic amino acid. Ayan. Okay. And then, sino pa? Lysine and si arginine. Ayan, mga basic amino acid. So, this is wrong. So, our answer is letter D. It can serve as a buffer at pH 7. Right? So, now, you may wonder bakit. Ayan. So, bakit kaya maaaring mag-serve as a buffer at pH 7? It's because of its isoelectric point. No? The isoelectric point of histidine is the only amino acid. No? Histidine is the only amino acid with an isoelectric point near 7, pH 7. So ibig sabihin, it can act as a buffer at pH 7, right? So the answer here is letter B. Yan. Next is yan. Number 58, which is the most acidic amino acid. Okay? Now remember, there are only two acidic amino acids. No? So you have aspartic acid and glutamic acid. So eliminate na natin yung mga hindi acidic. Okay? We know that asparagine is the amide of your aspartic acid and glutamine is the amide of your glutamic acid. Okay? Yan. So, dalawa na lang. No? Ito naman yung pinaka kailangan nyo maalala. Remember that aspartic acid only has two, no, carbons in the R group. Ito, R group na lang i-drawing ko ha. Ayan. So, sorry. COOH. No? Ayan yung R group ng aspartic acid. For glutamic acid, no, ang kanyang R group ay CH2, CH2. Ayan. Okay, there are 3 carbons. Okay? Kumpletuhin natin para hindi tayo nalilito. NH2, tapos H, COOH. Tapos ito rin, CH, NH2, COOH. Alright? Ayan. Now remember, no, according to organic chemistry, the longer the alkyl chain between two carboxylic acid groups, ito yung dalawang carboxylic acid group, the lower is its acidity. Okay? So in other words, no, between the two, no, ang mas maiksi ang alkyl group in between the two carboxylic acid functional groups ay C aspartic acid. So that is our answer. Ayan. So our answer will be letter B, aspartic acid. It's the most acidic amino acid. All right? And number 59. Asparagine has pK1 and pK2 values of 2.02 and 8.80. Yan. Respectively. What will be its net charge at pH 1.0? Ayan. So, now pH 1 is always on the left side. It's always on the left side of both the pK1, 2.02 and 8.0. Okay? So, anong ibig sabihin nun? Laging protonated. Yeah, so the first proton, which is the carboxylic acid functional group, is protonated. So its charge is 0. Okay? And so remember, no, ang itsura kasi ng functional group na yan ay COOH. So when it is protonated, its charge is 0. Okay? And then, yung 8.80, this is the amino functional group. Okay? So NH3+. Okay? Now, its pK value is 8.80. pH 1 is on its left side. It's protonated. So, ito yun. No? Protonated. So, its charge will be positive 1. Okay? So, therefore, the answer is letter C. Okay? Positive 1. Ayan. Number 60. Ayan. Monosodium glutamate is the sodium salt of glutamic acid. It is a flavor enhancer often added to restaurant foods, canned vegetables, soup, deli meats, and other foods. The pK1, pK2, and pKr of glutamic acid are 2.90, 9.607, and 4.27 respectively. Which of the following statements are true regarding glutamic acid? Ayan. So number 1, catabolism of glutamic acid involves oxidative deamination of the acid, the amino acid, to alpha ketoglutarate acid and ammonia, which enters the urea cycle. This one is true. Okay, so we have already eliminated letter C. Kaya number 2, glutamic acid contains 3 titratable groups. No? Kanina ko pa dinadrawing glutamic acid. So most likely, dapat ngayon, gets na natin na yes, this is true. Ayan. So 2 is also true. No? Glutamic acid is an essential amino acid. Bakit parang wala ko na expected? True. Which is true. Okay, so which is true? Okay, so which is true? So number 2, ayan, glutamic acid contains 3 titratable groups. 2 is the correct answer. True. Okay. Number 3. Okay. Glutamic acid is an essential amino acid. So actually, no, ang pinaka sagot na lang is A. Pero tingnan natin, no, kung wala ba. Number 3, glutamic acid is an essential amino acid. Ayan. So remember si Henry Matil, Vice President. No? Ayan. So nandiyan ba si glutamic acid? No? Walang G. So walang G kasi remember in this acronym, no, only the first letters, no, of the amino acid are there. No? So for example, H is histidine, M is methionine, A is not alanine, this is arginine. The two T's, no? So you have threonine at saka si tryptophan, no? Ayan, not tyrosine, no? Kasi tyrosine comes from phenylalanine. I is isoleucine, okay? Tapos L, two L's, dalawa lang naman yung L's, leucine and lysine. V is valine, and P it's not proline, no, but it's phenylalanine. Okay? So wala dito si glutamic acid, so number 3 is wrong. Okay? So dahil 3 is wrong, mali na yung letter B. Okay? So mali na rin yung letter B. Kaya, pero tingnan pa rin natin. Number 4, Monosodium glutamate can be formed from glutamic acid in a solution with a pH greater than 9.67. Ayan. So, remember, no, that the pK values are as follows. 2.19 for the carboxylic acid proton, 9.67 for the ammonium proton, and 4.25 for the other carboxylic acid proton. No? Ayan. So, for this one, okay, um, if the pH is greater than 9.67, ibig sabihin, deprotonated ito, deprotonated din ito. Okay? So, it's not monosodium glutamate, but disodium glutamate. So, number 4 is wrong. Yan. So, feeling ko get na nila talaga yung explanation, no? Kaya they use the rule. And number 4, the isoelectric point of glutamic acid is 5.37, at which pH is its net zero is its net charge is 0. Ayan. So the isoelectric point, no, of glutamic acid can be computed, no, using the two lower pK values. No? Ayan. So 2.19 plus 4.25 divided by 2 is definitely not 5.37. So this is wrong. So our answer is letter A. Only 1 and 2. Okay. So next we have, ayan, number 61. Arginine is an essential amino acid containing the guanidine group. Which of the following, no, statements are true regarding arginine? Ayan. Um, Number 1, it has a characteristic strong absorbance at 280 nanometers. This is wrong. Kasi remember that only aromatic amino acids like tyrosine and tryptophan will absorb at 280 nanometers. Phenylalanine, no, although it is aromatic, will not absorb strongly at 280 nanometers. Number 2, it is considered as a glucogenic only amino acid. Ayan. So, for this one, no, ayan, remember na ang glucogenic only amino acids are the rest, no, except for PI, PPP. Ayan. Okay? Ayan. So, this one is actually correct. And then number 3, it is the immediate precursor of urea in the urea cycle. Arginine is cleaved by arginase, no, to produce ornithine and ammonia. No? Ayan. So it is indeed the immediate precursor of urea in the urea cycle. Number 4, it produces a red-colored complex with alpha-naphthol and hypobromide. Ayan, in Sakaguchi test. No? So sabi natin, Sakaguchi arginine. Okay? So this one is also true. Number 5, it produces serotonin as a specialized product. This one is produced, no? Serotonin is produced by tryptophan. No? Ayan. So this one is wrong. So our answer will be 2, 3, and 4. And okay. Let's proceed to number 62. And tyrosine, a non-essential amino acid, is derived from which essential amino acid? Ayan. So our answer is letter A, phenylalanine. Kasi they have the same aromatic functional group. Number 63, which is an essential amino acid? Ayan, dapat talaga umaalis, no? And 1, 2. Which is an essential amino acid? Essential, no? Henry Matil VP pa rin, no? Yung ating ginagamit dito, no? Of these, A is alanine. So this is not essential. Ayan, kasi the A in Henry Matil VP is arginine. L is an essential amino acid. No? Ayan, so L is an essential amino acid. Kasi nga, dalawa lang naman yung L, leucine and lysine, and both of them are in the acronym. Ayan. And then C, P is proline. No? So proline can be produced from glutamic acid. Okay? So this is not essential. Letter D, tyrosine, is derived from phenylalanine. So this is also not essential. The answer is letter D. Number 64. The nucleosome contains DNA molecules bound to histone proteins, which are rich in lysine residues. Which is the isoelectric point of lysine, given the following pK values. Lysine is a basic amino acid, so we will get the average of the two higher pK values. So our answer is letter B, 9.74. Number 65, at a pH equal to the isoelectric point, lysine will have a net charge of um, 0, letter B, because that's the definition of isoelectric point. No? It is the pH at which the net charge of amino acids would be equal to 0. Number 66, which is true about the peptide bond. Ayan. The peptide bond has a partial double bond character. It's true. Okay. Letter B, it is ionized at physiologic pH. No, that's wrong. Because it's a covalent bond. Letter C, it is cleaved by agents that denature proteins. Ayan, that's false. Denaturation of proteins only break your van der Waals forces, ionic interactions, no, and other non-covalent bonds. So letter C is wrong. Letter D, it occurs mostly in the cis configuration. Remember, too much steric hindrance occurs because of cis configuration. So trans ang kadalasan for the peptide bond, excuse me. Ayan, except for proline. No? Ayan, which is more stable in cis configuration. That's the reason why proline can be found in the beds, no, of your amino acid. So D is wrong, so only letter A is correct. Number 67, the sequence of amino acids in protein is called the letter A, primary structure. Number 68, which of the following statements about protein structure is true? Letter A, the information required for the correct folding of the protein is contained in the specific sequence of amino acids along the polypeptide chain. This one is true. Ayan. Letter B, proteins consisting of one polypeptide can have quaternary structure. This is false because multi-subunit proteins would have a quaternary structure. Letter C, the formation of a disulfide bond in a protein requires that two participating residues of cysteine should be adjacent to each other in the primary sequence of the protein. No, that's not true. No. Ayan. Because disulfide bridges can be formed, no, even if the cysteine residues are far apart. Ayan. So this can happen in both your alpha helical protein and also in your beta sheet. Letter D, the denaturation of proteins leads to irreversible loss of secondary and tertiary structure. This is again false, no? Because denaturation, in fact, is reversible. Okay? Ayan. So letter D is false. So our answer is letter A. Ayan. And that is called your antisense dogma. Number 69 to 70, study the structure of the peptide below, which amino acid can be found at the end terminus. Yan. So if we look at the structure, no, this is the carboxy terminus, and this is your amino terminus. No? Ayan. So nakabaliktad siya. Okay, so remember that in naming your peptides, you always begin with the amino terminus and then we end with the carboxy terminus. Ayan. Ilan na lang ba tayo? 53. We started with 50 plus. We are now only 53. Yeah, so thank you so much for those who are staying. No? I suppose those who left already know the answer. Ayan, okay. The amino acid that can be found in the amino terminus is your proline, letter C. Number 17, which is the correct name of the peptide whose structure is shown below. I think the reason why they are leaving because they know that this meeting is being recorded. No, I think that is not fair for those who are staying in this classroom. No? Ayan. So number 70, tingnan natin no, yung sulat natin yung mga amino acids no in the peptide sequence. Ayan. So again, this is the amino terminus and then this is the carboxy terminus. Ayan. So this is your proline, no? Ayan. So that's your proline. Ayan, ayaw sumulat. Palit tayo. That's proline. Tapos this one is your imidazole ring. So that's your histidine. Okay. And then over here is your alanine. And then this one is your lysine. Ayan. Ay, sorry. Hindi yan lysine. That's your arginine. That's your arginine. R. Okay. Ayan. So if we are to read the name of the peptide, so from the amino terminus to the carboxy terminus, that's prolyl, histidyl, alanyl, arginine. Ayan. And that is letter B. Okay? Ayan. Prolyl, histidyl, alanyl, arginine. Okay? Ayan. So the answer is letter D for number 7. Ayan. Okay, next we have number 71, which is the correct name of the peptide whose structure is shown below. Yan. So just like what we did in the previous number, first we identified the amino terminus and the carboxy terminus. No? So this is the amino terminus and this is the carboxy terminus. Yan. So in this case, tama yung order no, of the peptide no, from left to right. So the next thing that we will do is we will identify the peptide bonds. And so let's identify the peptide bond. This is a peptide bond. This is a peptide bond. This is another peptide bond. This is another peptide bond. This is another peptide bond. Another peptide bond. Another peptide bond. Another peptide bond. Okay. Now that we have identified the peptide bonds, it's now easy to find the alpha carbon. Okay. So the alpha carbon is the carbon where you will find your R group. Okay. So this is the first R group. This is the second R group. This is the third R group. This is another R group. This is another R group. This is another R group. Another R group. Another R group and another R group. All right. So now we can name the amino acids no, from left to right. Yeah. So the first amino acid is a sulfur-containing amino acid. It's a sulfide-containing amino acid. So this is your methionine. Okay. Now the second amino acid has 3 carbons. Okay. So pag 3 carbon pag 4 pala, sorry, 3 ang sabi ko. Pag 4 carbon-containing amino acid, konti lang sila, no? So you have your leucine, isoleucine, you have your lysine. Okay? Yeah, so yan lang yung 4 carbon-containing amino acids, at least na aliphatic. Okay? So among the branched chain amino acids, there are only 2. You have leucine and you have your isoleucine. And this one is your isoleucine I. Now, this is another 4 carbon-containing amino acid in the R group. So this is letter Y, no? Yan pa letter Y yung structure niya. This is your leucine. Another leucine. Ayan, tapos another isoleucine. This one is a sulfide-containing amino acid, no? A sulfur-containing amino acid. This is a thiol group. Ayan, so it's a thiol group. So that's just one. That's your cysteine. And then this one is 1, 2, 3, 3 carbons, carboxylic acid. This is your glutamic acid, letter E. No? And then this is 1, 2, and this is your asparagine. No? Yan, because there are 2 carbons. It's an amide no, of a carboxylic acid amino acid with 2 carbons. Yung 2 carbon containing amino acid no, in the R group, no, na carboxylic acid is only aspartic acid. So this one is asparagine. Okay, so asparagine is N. Tapos, the last one is 1, 2, 2 carbons, hydroxyl group, dalawa lang, yung mayroong hydroxyl group na aliphatic amino acids. You have your serine and your threonine. Pag isa lang yung carbon, that's serine. Pag mas matanda, threonine. Okay? Yan, dalawang carbon. Okay, so this is your threonine, letter T. So our answer is letter D. Ayan, so millicent. Ayan, so kung napansin nyo lang, no, ayan, it's actually adenine. So another way of answering this kind of problem is this. No? And now that you have identified the amino terminus and the carboxy terminus, no, try to count the number of amino acids no, that are present in the peptide, okay? Based on the number of our groups that you will observe. No? So 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9. Okay? So you look at the choices, you already eliminate those which do not contain 9 amino acids. Okay? So tanggal na si malikit. Ayan. Tapos millipid, 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 din. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Maleficent, mali na rin. Okay? Kasi 10 amino acids meron siya. So there's only a choice between the two, no? Millipede or millicent. Ayan. So pag nagsimula ka from left to right, okay, pareho silang milli. Okay? So medyo mahihirapan ka. So what you can do is just look at the last amino acid, okay, and identify kung sino sa kanila yon. Okay? So since this one is hydroxide containing amino acid, so that's your millicent, letter D. Okay? Number 72. Ayan. For number 72, in protein sequencing, which method utilizes 1-fluoro-2,4-dinitrobenzene or FDNB to identify the N-terminal residue of the polypeptide. Okay? So, ang important key term dito is identify N-terminal residue. Okay? Now, aside from fluorodinitrobenzene, no, dinyl chloride or dabsyl chloride is also used, no, in this method, and that is your letter B, Sanger's method. Okay, number 73. Which protease or reagent leaves at the carboxy terminus of the amino acid shown below? Ayan. So, we have to identify kung anong amino acid siya. Ayan. So, this amino acid is, ayan, this is your phenylalanine. Okay, ayan. So, this is your phenylalanine. Ayan, because of this, no? Phenyl, tapos this is your alanine. Okay? So, phenyl, ito yung alanine. Okay, so si phenylalanine. Okay? So, sabi dito, carboxy terminus daw of the phenylalanine is cleaved, and that is your chemotype. Letter A. And now, number 74, the protease trypsin is active in wet pH condition. Yeah, so trypsin is it's it's an enzyme, it's a protease that can be found in the intestine. It's an intestinal na protease. No? Yan. And this molecule is actually activated in the presence, or it is more active if the pH of the medium is basic. Okay? Kasi nga nasa intestine. Alright, letter B, basic. Okay, number 75. Which of the following substances, okay, yan, 1, 2, okay, which of the following substances is involved in the formation of trans-hydroxyproline and hydroxylysine during collagen synthesis? Okay, now remember na hydroxylation of proline and lysine are important in the synthesis of collagen. Okay, and this one requires the enzyme prolyl hydroxylase and lysyl hydroxylase. And that particular, those enzymes, no, require ascorbic acid. Kaya nga, diniscuss natin before na ascorbic acid is required, no, in collagen synthesis. And lack of ascorbic acid in the diet could mean brittle bones, no, yan, or faulty synthesis of collagen, leading to diseases, no, like Ehlers-Danlos syndrome, osteogenesis imperfecta, and even scurvy. Okay. Number 676. Elastin cross-links involve covalently bonded blank residues. The answer is, yeah, tama. Letter C, lysine. The cross-links, no, that are present in elastin ay tinatawag natin na desmosine. Ayan. Number 77. Which fibrous protein contains mostly hydrophobic amino acids as well as sulfur-containing cysteine and methionine. Now, ang key dito ay hydrophobic, cysteine, methionine. But before we proceed to that, no, ayan, review lang natin. No? Remember, there are basically two types of proteins. No? You have the globular protein and you have the fibrous protein. Your globular proteins are also known as spheroproteins, no? And they are the water-soluble proteins. No? Examples of them would include your albumin, no? Your globulin, no? Among others. No? And then fibrous proteins, these are insoluble. These are water-insoluble proteins, and they are also known as scleroproteins. Okay? Again, and among the fibrous proteins that we actually discussed, which are quite important, are the following. You have collagen, keratin, and elastin. Okay? Ayan. Myosin is again the protein that's present in the muscle. Ayan. Of these fibrous proteins, no, isa lang dyan yung maraming sulfur, no, and hydrophobic amino acids, and that is letter C, keratin. Okay? Ayan. So, number 78. Proteins that assist in the proper folding of large proteins is called letter, ayan, umalis na naman. Letter A, chaperonin. No? Molecular chaperones, also known as chaperonins, no, are barrel-shaped proteins, no, which assist in the proper folding of large proteins. No? Remember, in the previous number, we mentioned, ano number yon? In a previous number, we mentioned that letter A, no, the information that is required for the, ito, number 68, ayan, for the proper folding of protein is contained in the specific sequence of the amino acids, no? And that is called the Ankins dogma. No? While that is true, no, for larger proteins, it's quite difficult to spontaneously fold into the proper conformation. No? So, kailangan nila ng help from another protein, no? Which is the chaperonin or the molecular chaperone. Okay? So, para madaling tandaan, pag sinabi kasi natin chaperone, it's basically a person that you bring along with you. No? Ayan. So, chaperone. Okay? So, parang yun din yung purpose or function of your molecular chaperone. No? Number 79, proteins that tag damaged proteins, yeah, or improperly folded proteins to be degraded by the proteasome, leaving their peptide bonds, no, is called your letter B, ubiquitins. Ayan. So pag sinabi ninyong ubiquitins, these are proteins that are ubiquitous. Ayan. They identify improperly folded proteins, no, or those proteins which need to be recycled. Okay, and then they bring, no, they bring the protein in the proteasome, which eats up the protein. Okay, para ma-recycle yung amino acids, no, from the protein. Okay, kaya ang tinatawag natin sa system na yon is the proteasome ubiquitin or ubiquitin proteasome system. Okay, of degrading your protein. Number 80. Which one of the following statements about myoglobin is true? And letter A, heme is a porphyrin molecule with a centrally bound ferric ion held by the 4 porphyrin nitrogens. What makes this wrong? What makes this statement wrong is the ferric ion. No? Yan, remember that the heme is a porphyrin molecule with ferrous ion in the middle. Okay? Yan. So letter B, myoglobin consists of a single polypeptide chain containing a tightly bound heme prosthetic group. No? Ayan. Tingnan natin yung iba. No? Letter C. The interior of a myoglobin is composed almost entirely of charged polar amino acids. Ayan. So what makes this wrong is yung interior daw ay polar amino acids yung laman. That's wrong. Remember na when proteins fold, the hydrophobic proteins are in the middle at the interior portion, and then yung inyong aqueous or water-soluble amino acids are at the outermost portion. Okay? So letter C is wrong. Okay. Yan. So letter D. Each myoglobin molecule binds 2 to 4 oxygen molecules via the proximal and the distal histidine molecules in the interior of the polypeptide chain. So in fact, this is also wrong. Ayan. Ay, sorry. This, sorry, diyan wrong. Ay, ayan. Actually, only B and D, no, yung ating naiwan na choices. No? So ngayon, imagine, examine na natin kung alin yung tamang sagot. Okay? Now, if you recall what I mentioned last time, okay, the myoglobin and the hemoglobin are quite similar in structure. Okay? So the myoglobin has the alpha and the beta chains. Ayan, dalawa yung chains niya. Ayan, so alpha and beta chains. Okay? So ang ibig sabihin nito, hindi siya single polypeptide. Okay? Ayan, so hindi siya single polypeptide chain kasi there are 2 polypeptides. Hemoglobin, no, has 2 alpha and 2 beta. Okay, so yun yung pinagkaiba nila. Okay? Now, both myoglobin and hemoglobin can bind this number of oxygen molecules, 2 to 4 oxygen molecules in the center of the polypeptide chain. No? So the answer here is letter letter B. Ayan. So letter B, myoglobin, and let's just change. Each myoglobin molecule binds 2 to 4 oxygen molecules via the proximal and distal histidine. All right. Number 81. Ayan. The deoxygenated form of hemoglobin is the letter D, dog. Ayan. So that's the T form. Okay? Remember that the deoxygenated form is the tout conformation na tinatawag. No? Ayan. So siya yung tinatawag na tout conformation. Kaya sa T. No? Ayan, tout. Ayan. R means relaxed. No? The relaxed form is the oxygenated form of hemoglobin. And so number 82, the deoxygenated form of hemoglobin is stabilized by all of the following except. No? Yeah. So remember that the deoxygenated form of hemoglobin is stabilized, no, if number one, if the pH is high, no, or if the concentration of the H plus is also high. Ayan. Another thing that stabilizes the deoxy form of hemoglobin is the high carbon dioxide. Okay. Another thing that stabilizes, no, the deoxy form of hemoglobin is high 2,3 DPG. Yan. So yan na yun, no? Yan, 2,3 DPG. Okay. So if we look at the choices, A, 2,3 biphosphoglycerate, this stabilizes your deoxy form of hemoglobin. Carbon dioxide stabilizes your deoxy hemoglobin. H plus, no, or acidity stabilizes deoxy form of hemoglobin. So the only answer here is letter C, carbon monoxide. Remember that the carbon monoxide stabilizes the oxygenated form of hemoglobin. Anong ibig sabihin nun? Carbon monoxide forms carbon monoxy hemoglobin. Okay, ulitin natin yun. Carbon monoxide forms your carbon monoxy hemoglobin, no? Which binds oxygen strongly. Okay? In other words, no, if the hemoglobin, no, goes out of the lungs and into the tissues, the oxygen molecule is not transferred to the myoglobin. Okay? Bakaw, no? Hindi ipamimigay ng hemoglobin with carbon monoxide yung oxygen niya. Okay? In other words, carbon monoxide stabilizes the oxygenated form of hemoglobin. All right? So letter C is our correct answer. Number 83. Tingnan ko lang kung sino-sino yung mga umalis. Okay. Oh, kaya 58. And so number 83, which one of the following statements about the allosteric effectors of hemoglobin is true? Ayan. So let's begin with, ayan, letter A. A high concentration of 2,3-biphosphoglycerate in the erythrocyte decreases the binding affinity of oxygen to hemoglobin. Looks good, no? Ayan, looks good. Sige, tingnan muna natin ibang choices. Letter B. Carbon monoxide decreases the oxygen affinity of hemoglobin by binding to the oxyhemoglobin. Decreases the oxygen affinity, right, of hemoglobin by binding to the oxyhemoglobin. Okay? This one is wrong, no? Kasi sabi ko lang kasi kanina. Carbon monoxide increases oxygen affinity by binding to oxyhemoglobin. Yan. So this one is wrong. Yan. So B. Letter C. Oxyhemoglobin and the oxyhemoglobin have the same affinity for protons. This one is also wrong. Yan. Because the oxyhemoglobin is stabilized by protons. Okay? Letter D, yan, dalawa na lang yung choices natin, no? So, A or D. No? So, the binding of carbon dioxide stabilizes the Taut conformation of hemoglobin, or letter A, no? A high concentration of 2,3-biphosphoglycerate in the erythrocyte decreases the binding affinity of oxygen to hemoglobin. Okay, so the correct answer here is letter A. Ayan. So, Remember that because of 2,3-biphosphoglykoside, a high concentration of it, no, mas marirelease yung oxygen, no, into the myoglobin. All right? Okay. So next number 84. The methemoglobinemia can be caused by which of the following, each of the following. Letter A, deficiency in NAD8 cytochrome B5 reductase. Letter B, glutamic acid to valine mutation at position 6 of the beta globin chain. Letter C, glutamic acid to lysine mutation at position 6 of the beta globin chain. Or letter D, and imbalance in the synthesis of globin chains. Ayan. So, isa-isay muna natin. Letter D, imbalance in the synthesis of globin chains is thalassemia. Okay? Ayan. So, this one is your thalassemia. Thalassemia. So, hindi yan methemoglobinemia. Alright? Next is glutamic acid to lysine mutation. This is your crystalline hemoglobin or HbC. Glutamic acid to valine mutation is your this is your sickle cell anemia. Okay, so sickle cell anemia is HbS. Ayan. And letter A, deficiency in NAD8 cytochrome B5 reductase means that the ferric ion that is normally produced, no, if oxidants are present, can no longer be reduced back to the ferrous form. Okay? Kaya nagpo-proliferate, dumadami yung ferric ion in hemoglobin. Okay? Yan. So that means, no, nagiging siyang methemoglobin or HbM. Yan. So HbM is letter A indeed. Okay? And so A. All right. Number 85. Blank refers to the enzyme with its cofactor. Ayan. So remember that for proteins, no, ayan, for proteins, meron tayong tinatawag na apoprotein and meron tayong tinatawag na prosthetic group. Ayan. Now, this one only happens if you have a conjugated protein. Okay, ayan. So pag meron tayong conjugated protein, ako ilalagay. Conjugated protein. And meron tayong apoprotein, which is the amino acid component, yan, of the protein, and the prosthetic group, which is the non-amino acid component. No? Non-amino acid component, okay, of your protein, okay, which is usually covalently bonded on the apoprotein. Now, the apoprotein plus the prosthetic group is your holoprotein. Ayan. Whole, no? Whole protein, buo. Okay? Now, for enzymes, it's the same, no? Pag apoprotein, so meron kang apo enzyme, okay? Plus a prosthetic group, the prosthetic group can either be a cofactor or a coenzyme. Okay? Yeah, so remember, pag sinabi natin cofactor, it's inorganic. No? Pag sinabi natin coenzyme, organic. Okay? Now, apo enzyme plus a prosthetic group is what you call a holo enzyme. Okay? Holo enzyme. Normally, holo enzymes are active, no? But some enzymes, some enzymes require cleavage, no? Ayan, kailangan nilang ma-cleave, no? Doon sa whole protein. Okay, mababawasan sila. Okay? Yung mga proteins which are yung mga enzymes, no, which are complete, no, enzymes which are complete but they are inactive, they are called your proenzyme or zymogen. They are called proenzyme or zymogen. So for example, your trypsinogen, and trypsinogen is a proenzyme or a zymogen, no? Yan. Obvious naman, may gen sa dulo. Yan. So it requires cleavage of certain peptides, no, in the trypsinogen molecule for it to be activated, okay, into trypsin. Okay? Yan. So yan yung mga terms, no? Ayan, na you have to remember. Okay? So going back to the question, blank refers to the enzyme with its cofactor. So that is your letter C, holo enzyme. Okay? Ayan, kasi apoenzyme is just the amino acid component, peptide, no, in your enzyme. Wala siyang prosthetic group. Coenzyme is the organic, no, organic covalently bonded group, no, considered as a prosthetic group to the apoenzyme. And then zymogen, no, it's the proenzyme, okay, which is inactive. Oh, yeah. So it has to be activated. No? Yan. Number 86, trypsinogen is the inactive form of trypsin. This is called, ayan, so nasagot ko na rin yung tanong ko kanina, letter D, zymogen. Okay? Ayan. Actually, what activates trypsinogen is also trypsin. Okay? Ayan. Ba weird. Ayan. Number 87, to yield the active enzyme, the acidic propeptide from trypsinogen is cleaved by the protease called, ayan. So, yung protease, no, na ginagamit natin for your trypsinogen is actually your enterokinase, letter C. Okay? Number 88. Menkes disease is an X-linked genetic disorder characterized by a reduced lysyl oxidase activity. This enzyme cross-links tropocollagen into strong collagen fibrils. Which cofactor is required by this enzyme? So lysyl oxidase is or has no copper, ayan, as its cofactor. Remember in our discussion, no, na-mention natin doon that deficiency in copper results in Menkes disease. No? Yung Menkes disease, this one results in hypotonia. Ayan. So, nagre-result ito sa hypotonia or lack of muscle strength. Okay? Ayan. So, hypotonia. Tonia yung nasulat. So, hypotonia. No? Ayan. So, normally, babies with Menkes disease cannot stand. Okay? Ayan. If they live long. Okay? And then, let's proceed to number 89. And numbers 89 to 90, consider the conversion of L-tyrosine and L-tyrosine from L-phenylalanine that requires the enzyme phenylalanine hydroxylase as shown below. Ayan. Yeah, so this is the diagram. The number 89, which accurately describes the enzyme. It is a or an blank. All right? So tingnan natin no, kung ano nangyayari. The phenylalanine hydroxylase is this one. Ayan. So two oxygen molecules, no, two oxygen atoms are added, no, in the molecule in the substrate and H plus. Okay? Yan. So kung papansinin natin kung saan napupunta yung oxygen, okay? There are two oxygen atoms here. One oxygen is actually here. Okay? Saan napunta yung isa pa? Yung isa pang oxygen. Okay? Kung papansinin ninyo yung structure, no, na naiwan ito, that's basically this one. In other words, the other atom of oxygen went where? Napunta dito sa water. Okay? Ayan. So one oxygen attached onto the substrate, which is your phenylalanine. The other oxygen attached on H plus, forming water. Okay? And that kind of enzyme is what we call a that is what we call a mixed enzyme oxidase. No? Ayan, mixed enzyme oxidase, also known as a monooxygenase. Okay? Yeah, so it's a monooxygenase because it only adds one, no, oxygen. Okay, it only adds one oxygen on your substrate. Okay, from the atomic oxygen. All right. Pag sinabi nating dioxygenase, the two oxygen atoms should have been placed on the substrate. Okay. For example, si catechol, no? Ayan, yung catechol formation, okay, requires a dioxygenase. Okay. Hydrolase is an enzyme that catalyzes hydrolysis. Wala namang hydrolysis na naganap. Oxidase naman, pag sinabing oxidase, the oxygen is attached to the inorganic H2, okay, or H+. Okay? Yan. So, monooxygenase is also known as mixed function oxidase. Ayan. Number 90, in this reaction, tetrahydrobiopterin or H4 biopterin acts as, ayan. So for this one, tetrahydrobiopterin, which is this one, okay. So ito siya, ayan. Ang use niya is actually as a coenzyme. Okay, yan. It's the coenzyme for your phenylalanine hydroxylase. So, yan. Numbers 91 to 92. Consider the reaction below. Then hexokinase is the initial enzyme of glycolysis. It catalyzes the reaction, converting glucose to glucose 6-phosphate. And number 91, which accurately describes this enzyme? It is a or an. Ayan. So, ano ba yung nangyari, no? Tingnan natin kung anong nangyari. So, this is your ATP and this is the phosphate group. Your one phosphate group was transferred, no, from the ATP into the glucose molecule, forming this phosphate group. Okay. In turn, your adenosine triphosphate became ADP or adenosine diphosphate. So hexokinase is actually a type of letter D, and it's a transfer base. Okay. Yeah, so letter D is a transfer base. Ayan. Okay. So why is it a transferase? Kasi na-transfer. Ayan. So transfer niya yung phosphate group galing sa ATP papunta sa substrate. Okay? So sino pa hindi pa natin na-define dito? Ligase. Ayan. So pag sinabing ligase, it is an enzyme that is responsible for the formation of double bonds, no, by removing groups or removal of double bonds by the addition of groups. Okay? Ayan. Addition elimination enzyme ang ligase. Okay? Yung ligase naman, it connects. Ayan, it connects. Ayun lang. Two substrates. No? Ligase yun. Number 92, which is the correct cofactor of this enzyme. So remember that hexokinase requires magnesium. Okay, yeah. So nandun yun sa table, no, which I presented to you last time. Number 93. Yan, glucose 6-phosphatase is an enzyme involved in gluconeogenesis. It produces glucose from glucose 6-phosphate. Ayan. Which of the following is true regarding this enzyme? Ayan. So, tanda na, glucose 6-phosphate is the substrate and what you form is glucose, no? With the use of glucose 6-phosphatase. So, ano yung nangyari sa kanya, no? Ayan. Tingnan muna natin letter D. And it is a phosphorylase enzyme that transfers the phosphate group from a substrate, resulting in breaking of bonds between its monomeric units. And remember na, this phosphorylase enzyme usually, no, usually breaks bonds. Okay? Ayan. Wherein monomeric units will be produced. Okay? So magda-add ng phosphate group, no? Ayan. So for example, this is polymeric, no? Let's say si glycogen. Okay? So magdadagdag siya ng phosphate. Yan, magdadagdag siya ng phosphate sa kanya, okay? In turn breaking this bond. Okay? Ayan, so ganyan yung function, no, ng inyong phosphorylase enzyme. Okay? Now, that's not what happened, no, with glucose 6-phosphatase. So letter D is wrong. Ayan. Letter C, it is a synthetase enzyme that catalyzes the transfer, ayan, of phosphate group to an organic substrate via high energy ATP. This is definitely wrong. No, ayan. Kasi baliktad. No? Ayan. So remember na ang start mo is glucose 6-phosphate naging glucose. Okay? Natanggalan ng phosphate. Okay? Remember that synthetase enzyme, no, requires ATP. No? And in turn, no, ay normally nadadagdag din yung ATP into the substrate. Okay? Ayan. So hindi siya natatanggal. Okay? So C is wrong. Letter B. It is a kinase that catalyzes the transfer of a phosphate group from ATP to an organic substrate. This is again wrong kasi baliktad. No? Ayan. Hindi tayo nadagdagan ng phosphate. In fact, natanggalan tayo ng phosphate. Kasi sabi dito, it produces glucose from glucose 6-phosphate. So B is wrong. So the answer is letter A. No? It is a hydrolase enzyme removing phosphate group from a substrate using water. All right? Yan. Next number, 94. Yan. So we are now in enzymes. Yan. The Michaelis-Menten constant, Km, is letter A, numerically equal to 1/2 Vmax. Letter B, dependent on the enzyme concentration. Letter C, numerically equal to the substrate concentration that gives maximal, half maximal velocity. Or letter D, independent of pH. All right? So now remember no, that the Km, ayan, remember this drawing, ayan, this is the substrate concentration in the x-axis, and this is the velocity, the initial rate no, of the reaction. Now, normally, no, for enzymes, as you increase the substrate concentration, the rate of the reaction also increases. Okay? And from here, we can actually determine the Vmax. No? Ito yung Vmax. Yan. Okay? Now, at half the maximum velocity, which is this one, right, we can determine a substrate concentration. Okay? Yan. So, at half maximal velocity, we can determine a substrate concentration we call Km or Michaelis-Menten constant. So, our answer is letter C. Numerically equal to the substrate concentration that gives half maximal velocity. Okay? This actually tells us the affinity of the enzyme to the substrate. In fact, the higher the Km, the lower is the affinity of the enzyme to its substrate. All right? Now, letter A is wrong kasi nga, it's not numerically equal to 1/2 Vmax. No? It's equal to the substrate concentration at half Vmax. All right? Letter B, dependent on enzyme concentration. And remember that this is a physical property of your enzyme. No? Kasi nga, the higher the Km value, the lower is the affinity of the enzyme to the substrate. So it's not dependent on enzyme concentration. And then independent of pH is also wrong kasi of course, enzymes no, are highly affected by the pH. No? And because of that, the affinity of the enzyme no, also varies with the pH. No? Pag in-extend pa natin yun, ibig sabihin yung Km, the affinity of the enzyme, no, is also affected by pH. All right? So number 95, the rate of an enzyme catalyzed reaction was measured using several substrate concentrations that were much lower than Km. The dependence of reaction velocity on the substrate concentration can be best described as, no, ayan, so sino sa kanila yung ating sagot? Ayan, teka lang, hinga lang ako saglit. Okay, so what is our Michaelis-Menten equation? And so that VO equals Vmax and times substrate concentration S over Km plus S. All right? That's our Michaelis-Menten equation. So sabi dito, no, if the rate of the chemical reaction or the enzyme reaction is measured using several substrate concentrations much lower than the Km, so ibig sabihin, S is much lower than the Km. Okay? Ayan. Ano na yung mangyayari? Okay, so if the S is much lower than the Km, then what happens is the S here becomes negligible. Okay? So kung negligible siya, it's as good as it does not appear there. No? Yan. So ito lang yung ating equation. Okay? So na-eliminate na natin yung substrate concentration in the denominator. Okay? Of course, it will not have any effect on the numerator. Okay? Ayan. So hindi natin ito tatanggalin. Okay? Now, normally, no, when you see something like this, ang agad-agad na isipin ninyo, this is the Y, this is the X. The X, this is the Y. So, ibig sabihin, this is the slope M. Okay? Y equals mx. Okay? So, in other words, okay, the velocity, VO, is directly proportional to the substrate concentration S. Okay? Ayan. So, yan yung makukuha natin from this equation. So, let us look at which of these ay tamang sagot. Letter A, it is a constant function of Vmax. This is wrong. Okay? Kasi the relationship is this. Okay? So, this is wrong. It's not a constant fraction of Vmax. Okay? Letter B, it's equal to Km, also wrong. Hindi naman yung pinapakita ng formula na nasa right side. It's independent of enzyme concentration. This is also wrong. Ayan. Of course, as you increase the enzyme concentration, the velocity should increase as well. And the only answer is letter B. Okay, ayan. So letter D, directly proportional to the substrate concentration as seen, no, in this in this figure. All right? Ayan. So yan yung ating Michaelis-Menten equation and yung initial rate of reaction. Right? Now, let's proceed to numbers 96, 97, and 96 and 97. And siguro wag ko na lang muna burahin yung mga nakasulat dito. No? Yan, i-retain natin siya. Gagamitin natin kasi yan. Okay, consider the data from the Lineweaver-Burke plot of an enzyme substrate reaction. The plot exhibits a slope of 5 times 10 to the negative 4 m times minute per mole and y-intercept of 0.1 minute per mole. All right? So for this one, remember that the Michaelis-Menten, siguro lipat pala ako. No? Ayan, para mas madali. Ayan. Lipat ako sa whiteboard. Hold on. So medyo masikip pala yung space. Ayan, I just realized. Kaya bumuka. Ayan. So let me erase everything. Okay. Ayan. So remember na this is your Michaelis-Menten equation. No? VO, Michaelis-Menten equation. And this is your VO equals Vmax times S over Km plus S. All right. Now, the Lineweaver-Burke equation, balik na. The Lineweaver-Burke equation is just the inverse of your Michaelis-Menten equation. No? So, 1 over VO equals, balik na rin din natin yung denominator, Km plus S over and Vmax times S. Alright? So now, when we rewrite this one, so this one becomes Km over Vmax times S, ayan, times S, gaya muna, plus S over Vmax times S. Alright? Now, according to your teachers in math, no, we cancel this out. Tapos, we can now rewrite the equation 1 over VO equals Km over Vmax. Tapos, ihiwalay ko lang yung 1 over S dito. Ayan. Plus 1 over Vmax. Ayan. So, what do we observe now? This one is already Y equals MX plus B. This is already equation of the line. All right? Wherein the slope of the line will be equal to Km over Vmax. Ayan. And then the y-intercept is 1 over Vmax. Now, if we compute for the x-intercept, the value of x when the y is 0, okay, ano yung gagawin natin? No? So, the value of x when the y is 0, so 0 natin yung y, so Km over Vmax. Yan, para hindi tayo malito, gawin lang natin x muna ito, plus 1 over Vmax. So this one, no, we transpose the values. So we have Km over Vmax times x equals negative 1 over Vmax. All right. Then we cancel Vmax. So x-intercept is equal to negative 1 over Km. And so that's another important, no, A lang yung ano natin dyan, symbol. A is equal to negative 1 over Km. All right. So these are the important values, no, in Lineweaver-Burke equation. Okay. So the slope is Km over Vmax. The y-intercept is 1 over Vmax. The A is negative 1 over Km. Okay. Yeah. Now, if we are to draw the plot for the Lineweaver-Burke equation, it would look like this. Okay. Yan. So remember that our y values ay 1 over VO, our x values ay 1 over S. Okay? Tapos we draw our line. Okay? So the slope of the line is km over Vmax. Ay, sorry. Mali ako nang na-point. The slope of the line, m, is km over Vmax. Ayan. Km over Vmax. And then, the y-intercept, yan yung tinuturo ko dito. The y-intercept, that is your 1 over Vmax. Ayan. And then, yung x-intercept, itong tinuturo natin dito is negative 1 over km. Alright? So, yan yung mga important features of your line-Weaver-Burke plot. Okay? Ayan. So, let's go back to the question at hand. Ayan. So, for this particular question, yan, magbura lang. Ay, hindi. Yan, magbura. Ayan. So, what is the Vmax of the reaction daw, no? Ayan. So, what is the Vmax? So, where do we get the Vmax? Ayan. So, remember, balikan, i-rewrite natin dito yung mga nalaman natin from deriving the equations, no? From the Line-Weaver-Burke equation. So, remember that the slope m is equal to km over Vmax. Tapos, the y-intercept b, ayaw sumulat, why is that? The y-intercept b is equal to 1 over Vmax. Okay? Now, we are given the slope, which is this one, and we are also given the y-intercept. Okay? So, the y-intercept is 0.1 minute per mole. Okay? And that is this b. No? So, 0.1 minute per mole. Okay? So, from there, we can already compute the Vmax. Vmax is just inverse of 0.1, which is equal to, so 1 over 0.1, that's equal to 10. Okay, 10 moles per minute. And that's our Vmax. Okay, so the answer is letter, and the answer is letter C. 10 moles per minute. Okay, then number 97, what is the Km of the enzyme? Yeah, so for the Km, ano naman yung ating gagamitin? Ayan, so we already know the Vmax and we know the slope. And ito yung slope, ito yun. We know the Vmax, so Km na lang yung kulang. Okay, so dito natin siya kukunin. The slope is 5 times 10 to the negative 4 minutes, mole minute, sorry, ulitin natin yun. Molar minute, m, minute per mole, okay, equals, and Km, which we don't know, and so Km over and Vmax, which is 10 moles per minute. Okay? So, para hindi nakakalito, dito ko na lang isulat, ha? Ayan. So, pura tayo. Para isang linya lang siya. So, this is 5 times 10 to the negative 4 molar minute per mole. Okay? So, Km is simply, mumultiply mo siya doon. So, Km is equal to 5 times 10 to the negative 3 moles. No? Magka-cancel kasi yung mole per minute. No? Ayan. So, we have letter C. Ayan, letter C. Letter C. Okay? Nag-get yan? Okay? Ayan. So, next number 90. Ayan, 98. Ayan. So, number 98, a competitive inhibitor of an enzyme. No? Ayan. So, tingnan natin no, kung naalala niyo yung code na ano ko sa inyo. So, so there are two ways to answer this. No? So, alalahanin natin yung mga graphs. No? So, remember, for competitive inhibitors, ganito yung ating graph. Okay? Ang Lineweaver-Burke equation. This is the uninhibited. Tapos, ito yung inhibited. Okay? Ayan. So from this diagram, what we see is that the y-intercept doesn't change. Okay? Ayan. So sino ba yung y-intercept? Diba? The y-intercept B, no? The y-intercept B is equal to 1 over Vmax. So in other words, the Vmax, no? The Vmax doesn't change. No change. Okay? And then, if you notice, no, yung ating x-intercept, ito yun, ay nagbago. Okay, yan, gumalaw. Okay, if there is an inhibitor. So the x-intercept A is equal to negative 1 over Km. Okay, because there will be an increase in the Km. Okay, Yan. Increase in the KM, anong ibig sabihin nun? As you add a competitive inhibitor, no, the affinity of the enzyme to the substrate decreases. Okay? Because the KM increases. Okay? Ayan. So, yan yung dito, no? KM increases. That's for competitive inhibition. Alright? Yan. So, remember, no, na you can use both the graph, no? Representation, and also itong code na binigay ko sa inyo last time. Okay? So, KM and Vmax, no? Alphabetical lang yan. K and V. CUN, no? Competitive, uncompetitive, non-competitive. Pag uncompetitive, parehong low ito. Tapos kapag non-competitive, this one is no change. This one is lower. Okay? So, hanapin natin yon, no? For competitive inhibition, so remember, pag competitive, di ba X, yung sabi ko, no? Last time, X. Kaya pa X ito. There is an increase in Km without change in Vmax. So, increase in Km without affecting Vmax. So, our answer is letter B. Alright? Ayan. Okay? So, clear tayo doon, no? Dali lang, di ba? Pag-memorize natin. Number 99, the presence of a non-competitive inhibitor. Ayan. So, ito naman, non-competitive. So, for non-competitive inhibitor, remember yung ating drawing for this is like this. Non-competitive, letter N. So, for non-competitive, ayan, this is yung without competition. Okay? Kapag mayroong competition, so kasi nga non-competitive siya, letter N. Ganito. Okay? So, ang pinamemorize ko sa inyo, isipin nyo lang parang letter N. Okay? Yan. So, ano yung mga nagbago? Okay? So, take note that the x-intercept did not change. Okay? So, non-competitive, no change in the Km. Kasi the x-intercept is -1/Km. Okay? No change in the Km. Pero, what happened with your Vmax? Remember, sa y-intercept, nagbago siya. Okay? And y-intercept is 1/Vmax. Okay? So, as we go up, bumababa yung Vmax. So, there is lower Vmax. Okay? So, hanapin natin yung answer na yon in these choices. There is no change in the Km, but there is a decrease in the Vmax. So, letter B. Okay? So, there leads, no, the presence of a non-competitive inhibitor leads to a decrease in the observed Vmax without affecting the Km. Letter B. Alright? Yeah, so, last number. Ayan, so alam ko yung iba excited. Kanina pa nga umaalis yung iba, no? Ayan, so number 100. 2,3-biphosphoglycerate and binds to the central compartment of the hemoglobin tetramer, and it stabilizes its south conformation because it decreases hemoglobin's affinity for oxygen. It is a or an blank of hemoglobin. Ayan. So the answer here is allosteric. Ayan. It's an allosteric inhibitor. Okay? Ayan. Of your hemoglobin, no? Kasi it decreases, no, the affinity of hemoglobin to oxygen. Okay? So, it's an inhibitor. Yung ibig sabihin natin ng allosteric, no, if you recall what we discussed last time, ganito lang yun, no? If this is your molecule, let's say your hemoglobin, normally the substrate binds at the active site, no? So, this is what you call the active site of the protein. No? So, let's say enzyme. No? Ayan, that's the active site. Okay? So, if you have a competitive inhibitor, the competitive inhibitor, no, let's say ito yung competitive inhibitor, binds also on that same active site. Okay? Now, there are certain molecules, no, which do not bind at the same site. Okay? So, for example, meron ditong site on the opposite side of the molecule. Tapos, doon ko connect yung isang molecule. Okay? Ayan. So, ang tawag natin sa site na ito is, ayan, so pangalanan na natin, no? So, this is what you call the active site, and this is what you call the allosteric site. Okay? It is a site outside or far from the active site. Okay? Now, attachment or binding of a substrate into the allosteric site can change the conformation of the active site. No? Either to, number one, allow the substrate to bind to that site perfectly. Okay? So pwedeng pag nag-bind itong kulay violet sa allosteric site, pwede niyang i-improve, no, yung fit of the substrate to that active site. Okay? So pag ganun yung nangyari, then we call that molecule an allosteric activator. Okay? Now, if the binding of this molecule reduced the affinity of the substrate to that enzyme or to that protein, okay? Ibig sabihin, no, imbis na pumasok yung substrate dun sa active site, hindi na siya makapasok. Okay? That is your allosteric inhibitor. Okay? So that being said, your 2,3-biphosphoglycerate, no, which binds on another area of your hemoglobin, is considered as an allosteric inhibitor. Letter B. All right? So that's it. Ayan. So we are done. No? Ayan. So are there any questions so far? Ayan. With all the discussions. Ayan. So we ended up with, ayan, ilan to? 58 participants. Oh, ayan. All right. So I think walang tanong. Okay. But still, no, ayan, some of you actually messaged me. No, ayan, those from University of Pangasinan. May mga nag-message sa akin over the past weekend, no, and during the past week. Tapos I already replied to you, and I hope no that you were able to use yung aking handouts, no, ayan. And again, no, I would like to request you not to upload the slides in Course Hero, SlideShare, or whatever, no. Ayan, kasi that would be quite not good. That would not be so good. Ayan, so yun lang, no, kasi nagiging available siya to everyone. No? Ayan, so yun lang yung request ko sa inyo. But if you have additional questions, no, you want to clarify anything else, no, do not hesitate to message me through my email address. Ayan, so some of your friends already know my email address, but just in case, no, you still don't know my email address, ayan, it's igfredobimata@gmail.com. All right. So with that, again, once again, thank you so much, everyone. No, have a happy weekend ahead. No, I'm not sure kung meron na kayong klase for tomorrow ulit. Okay? Yeah, but whoever goes there, no, I hope you give them the same amount of attention. No? Ayan. So maraming salamat po